Assalamu alaikum, everyone. I am Almazroua, a third-year medical student. And for today's session, inshallah, I'll be explaining ear anatomy and the physiology of hearing and balance, okay? So starting with the anatomy of the ear is a sensory organ that has two main functions. The first function being the hearing, and the second being maintaining the balance and equilibrium. So when it comes to the ear, when studying the anatomy, the ear is divided into three major parts: the external ear, which is this blue structure here, the middle ear, which is the yellow, and the inner ear. Just add my pointer. Okay. So yeah, so we said we have the outer, the middle, and the inner ear. So we're going to start by talking about the external ear. Now, the external ear is the first part is the part that's visible, part of the ear that we can see where we put our earrings. So this part is what we refer to as the pinna or the auricle. Now, looking at this is a picture of the right ear. So we have this part here, which is the helix, and then we have the antihelix. Then we have the tragus, antitragus, the lobule. I don't think they're going to ask you about the different parts of the pinna, but it's good to know them. If you look at the pinna or the auricle, you're going to notice that it looks like it has a very tiny tunnels, like looking at the anatomy of it, like kind of tunnel, and that's literally the function of the external ear. So it receives the sounds and it tunnels them all the way to the external auditory meatus. And the external auditory meatus would be the second part of our external ear. So the external ear is only composed of two parts: the auricle or the pinna, which is the part that's visible to us, which tunnels the sounds all the way until the external auditory meatus. Now let's talk about the external auditory meatus. So the external auditory meatus is made of fibrocartilage. So we know that our ear is made of cartilage. It kind of dips inside and forms the outer 1/3 of the external auditory canal to be formed again from cartilage. However, the inner 2/3 of the external auditory canal is made of bone, or the tympanic part of the temporal bone to be exact. So the outer 1/3 is made of cartilage, the inner 2/3 is made of bone. Another important thing about the external auditory canal is that we have skin overlying it. And this skin, just like any other skin, it has hair follicles and glands. But one specific type of glands that is important to us is the ceruminous gland, because these types of glands produce the earwax. And the earwax is important when it comes to protecting our ear from from water. So it waterproofs our ear. And the second important function is that it blocks any entrance of foreign bodies or insects or bacteria into our ear. So the wax of our ear has a very important protective function and is produced by the ceruminous glands. Now we're going to move to the nerve supply to the external ear. Now, before I explain the nerve supply, I just want to say that I added this picture, and it doesn't have the exact same nerves that Dr. Yaqeen talked about. I just added this picture so that you can kind of visualize where the nerves are at. But when it comes to taking which nerves to add to your exam or answer, then follow the ones that are written here, not the picture. Okay, so the auricle's outer surface, and he means this part of the ear, is innervated by the auriculotemporal nerve. And then the inner surface of the ear, which is this part, is innervated by the greater auricular and lesser auricular nerves that are coming from the cervical plexus. And then near the external auditory meatus, which is this area, it's innervated by, again, the auriculotemporal nerve. And there are other contributions from the tympanic branch of the vagus and the facial nerve branches. So multiple nerves are innervating this area. But again, I just added this picture so you kind of can visualize where the nerves are present at. Okay. So at the end of our external auditory canal, we have this structure here, which is called the eardrum or the tympanic membrane. So now we're going to discuss the tympanic membrane, which has very important findings. So if you've ever been to a doctor and he has placed the otoscope in your ear, they were trying to visualize the tympanic membrane. So this is what a healthy tympanic membrane looks like. It has different parts. First, it's present in the annular groove of the temporal bone. So that's where the annulus fibrosus is, or the annular groove of the temporal bone. Further in the lecture, I'll talk about the middle ear, and it has different bones. One of the bones that are present is the malleus. It's the first bone in the middle ear. And because the tympanic membrane, it is what's separating the external ear from the middle ear, we can see the first bone that is present in the middle ear, which is the malleus. So this here, the HM, this part here, is the handle of the malleus. And the place where it's attaching to the tympanic membrane is called the ombo, and that's where the ombo is. We also see here a cone of light, and this is very important, and it indicates the tympanic membrane is intact and it's healthy. If you don't see the cone of light, then this indicates that it's unhealthy. I think my internet connection is interrupting, so I'll just switch to my personal hotspot. Here, it's kind of explaining it clear. So this is the tympanic annulus, which is where the tympanic membrane is present. Here we have the malleolar folds. This is the pars tensa. The pars tensa is the part that is very rigid or dense connective tissue that is present in the lower parts of the tympanic membrane. And then we have the pars flaccida, which is flaccid and floppy, and is present in the upper parts of the tympanic membrane. Here we have the ombo, which is where the handle of the malleus is attached, and that's the handle of the malleus. Okay, now this is the chorda tympani nerve. We know that the chorda tympani is a branch of the facial nerve. I'm not going to go into details of this because you've had a full lecture about the cranial nerve 5 and 7. I'll just mention that the tympanic membrane, it courses from all the way in the eardrum. So it's sometimes visible while performing an otoscopy. Okay. Okay, now there's an important procedure that's sometimes performed in cases of emergency. So a very common infection in children, especially, is otitis media. And otitis media is inflammation of the middle ear. Now, sometimes, because we know in cases of inflammation, we have lots of fluids, lots of pus that is being produced. Sometimes the middle ear, it causes bulging of the tympanic membrane so much that the doctor fears that this tympanic membrane might rupture. So what the doctor can do is a procedure that is called myringotomy. Now, myringotomy is when they perform a small incision in the tympanic membrane to drain it, to remove the pus and the fluids, and this prevents the tympanic membrane from rupturing. So it's just a small incision in the posterior inferior aspect of the tympanic membrane. And it's very important to see the concave and the shiny appearance of the tympanic membrane and any landmarks. If you can see a landmarks in a tympanic membrane, this generally indicates that it's a healthy tympanic membrane. If you don't see the landmarks, then it's kind of because the tympanic membrane is bulging and sometimes can indicate that it's an otitis media. So this is just what I said. We're going to just go over the nerve supply. So the main nerve that is innervating the tympanic membrane is the auriculotemporal branch of the mandibular division of the trigeminal. And then there is also the tympanic branch of the glossopharyngeal nerve, also known as Jacobson's nerve, which forms the tympanic plexus in the medial wall of the tympanic cavity. I'll talk about this more in details in the later slides. And then we have the auricular branch of the vagus, also known as Alderman's nerve. Now, this nerve is very important in children and elderly because we know that the vagus carries parasympathetic fibers generally, like it's a parasympathetic nerve. So when the doctor is performing otoscopy, we kind of need to be careful when it comes to children and elderly because stimulation of this nerve, it can induce nausea and vomiting in children. However, in elderly, it can cause bradycardia, hypotension, and even syncope. That's why this nerve is to be considered or to be in the back of our heads whenever we're performing an otoscopy. Now, again, they're just the landmarks. We're just going to go over them. The tympanic annulus, it's where the tympanic membrane is attached. It's in the tympanic parts of the temporal bone. Here we see the pars tensa, which is the made of dense connective tissue. And that is the pars flaccida. Malleolar folds. This is the chorda tympani, which is a branch of the facial nerve. And then we have the ombo, which is the parts where the malleus is attached to the tympanic membrane. We have the handle of the malleus. And then we have the cone of light, which indicates the light reflex. And then we have the vascular and nerve supply that is going to the tympanic membrane. Okay, we discussed this already. So yeah. Now these are the different areas where the tympanic membrane can possibly rupture. So it can either be a very large perforation in pars tensa, or a smaller perforation in pars tensa and a perforation in pars flaccida. Now, spontaneous perforation can also occur in cases of acute otitis media that we discussed because of the accumulation of discharge pus. This can literally cause the eardrum to be bulging to the point that it cannot stand the amount of swallowing that is present, so it ruptures. Now, barotrauma can happen, and this is particularly important in children who have sinusitis. We'll discuss it later when we were talking about the middle ear, but generally, I think it's common knowledge that a very common presentation of upper respiratory tract infection in children is that they present also with otitis media. Their ear starts to hurt. I don't know if any of you have experienced it, but I personally did. When I was younger, I had an upper respiratory tract infection and my ear would hurt me and I would have otitis media. It's very common in children. However, the problem here is that there is a connection between the sinuses and the middle ear. In case of sinusitis, because again of the inflammation and discharge, this connection would kind of be blocked. So this can increase the pressure in the middle ear because the connection is blocked, and this can lead to perforation. Now, in relation to barotrauma, this is why people with sinusitis are not advised to travel on planes, because they increase the pressure. There is barotrauma in planes in general. So barotrauma, which increases furthermore pressure in the middle ear, this can increase the risk of rupturing of the tympanic membrane. That's what it's trying to tell you, basically. Okay. Now we're going to move to the bulk of the anatomy, which is the middle ear. Someone is asking me, could you please repeat what is the exact meaning of barotrauma? Barotrauma, let's break the word together. Baro means pressure, and trauma is just any traumatic injury. So barotrauma, whenever there is increase in pressure, this is commonly seen in planes. So when you travel in plane, when your ear starts hurting you because you're going upwards. So barotrauma is kind of a traumatic injury due to the high pressure of the planes. That's what it's trying to tell you. Did you get it? Okay, perfect. So now we're going to move to the middle ear. Now, one very important part of the middle ear to know is the borders. So we have different borders of the middle ear. The first one is going to be the lateral border, which should make a lot of sense to us, which is the tympanic membrane, because we said what's separating the external ear. Let's just visualize it. Okay. Oh my God. Yeah. What's separating the external ear from the middle ear is the tympanic membrane. So the lateral wall of this middle ear, it makes total sense for it to be the tympanic membrane. I want it to make sense to you so that when you're studying the middle ear, the borders, it doesn't overwhelm you. So the lateral border is the tympanic membrane, and it makes total sense. Now we're going to move to the superior border. It's made of a very thin bone that is called tegmen tympani. Now this bone is what's separating the middle ear from the middle cranial fossa. And this is important because in cases of infection of the middle ear, again going back, which is called otitis media, this infection can travel through the tegmen tympani and go to the brain. Now, in the brain, we have different meninges. And I'm sure you remember from neuro, what was the outermost meninge? Can someone tell me? The layer of the meninges? The outermost one. The dura, exactly. So when the infection travels to the dura, travels to the middle cranial fossa, the first thing that we'll encounter is going to be the dura mater. So now it starts to make sense with it. It can either cause an epidural abscess, so the infection did not really invade the dura yet, it's just confined outside the dura mater, or it can cause meningitis because it can cause infection and inflammation of the dura and maybe even the rest of the meninges, or it can reach the brain, causing a brain abscess. Or because we know in the dura we have different dural venous sinuses, it can also cause thrombosis of the dural venous sinuses. So these are all complications of otitis media in case it travels through the tegmen tympani. Now we're going to move to the floor or the jugular wall. Now the middle ear is kind of sitting on top of the internal jugular vein. That's why we refer to it as the jugular wall, which is the floor of it. Now, there are different nerves, again, present here, but I'm not going to go into details because you're going to take them in different lectures. I'll focus on the ear in this lecture. However, I'll talk about only one nerve. Now, the posterior wall is the mastoid wall. Now, this is really important. Remember when I said there is a connection between the middle ear and the sinuses anteriorly? Now, posteriorly, we have connection to the mastoid wall. The mastoid. The bone that is right behind your ear, that's what we call the mastoid bone. and it has air cells inside it. Now, any infection in the middle ear can travel to the mastoid and cause mastoiditis. That's why it's very important in children who have otitis media to check behind their ear to check if they're having mastoiditis or not. Now, this will the infection from otitis media, if it's traveling through the posterior wall, it will travel through a structure that is called the adytus. Okay, and then we have the anterior wall. The anterior wall is kind of formed because the floor and the roof are kind of joining, and it's going to form the anterior wall, and it's mainly formed like by the internal carotid artery. And another important structure is the eustachian tube. I just kept telling you that there is a connection between the ear and the sinuses. Now, this connection is through what's a structure that is known as the eustachian tube. So that's the eustachian tube, and we have the tensor tympani muscle. We'll talk about it later. Just know that it's also present in the anterior wall. Majority of the borders, it's just pure memorization. So you kind of, I'll help you as much as I can, but you kind of have to set and memorize them. If you kind of relate it to the other structures throughout the lecture, you will kind of understand the orientation. But generally speaking, it's pure memorization. Now, the medial wall or the labyrinthine wall, it's like, now think about it. We have the middle ear. Now, the medial wall is going to be the closest to the inner ear because we had the external, middle, and then inner. So the medial wall is going to be like what's right after it's going to be our internal ear. Now, in the internal ear, we have a structure called the cochlea. We're going to see it later. And the cochlea is kind of a snail-like structure or circular structure. And this is why it's going to form kind of a bulging in the medial wall of the middle ear. The oval window and round window, we're going to discuss them later. We also have the facial nerve present. It goes from the posterior wall all the way to the anterior wall. So as I mentioned, I won't go through the nerves in details, except it's one nerve. And okay. Now, the cochlea that I just talked to you about, which is present in the inner ear, it forms a structure that is called a promontory in the inner ear. And simply the bulging of the medial wall of the, in the middle ear. It's the bulging of the medial wall of the middle ear. It's caused by the cochlea. So it's going to form the promontory. And then there's another bulging that is formed by the facial nerve that is also present in the medial wall. Now, there's an important plexus that is present in the middle ear, which is called the tympanic plexus. Now, this tympanic plexus is formed by the glossopharyngeal nerve. So we have the glossopharyngeal nerve that goes to the, it's going to give a branch actually, that is called the tympanic branch of the glossopharyngeal nerve. Now, as the tympanic branch enters the middle ear, let's imagine that this is the middle ear, it's going to form a tympanic plexus. Okay, so let's say this is a plexus. Now, this plexus is sensory because it's important for like the sensations in the ear, but it's also carrying parasympathetic fibers. Now, this tympanic plexus is going to give a nerve that is called the lesser petrosal nerve coming out of the tympanic plexus. This lesser petrosal nerve will go all the way to the otic ganglion, which is supplying the parotid gland and contribute to the parasympathetic supply to the parotid gland. I think you also took this somewhere else, but I'm not sure if you took the part of the tympanic plexus, but this part is important and is frequently asked. So know that part of the glossopharyngeal nerve, it's what's forming the tympanic plexus in the middle ear. Okay, and that's basically it. Now we discuss the borders. Does anyone have any questions about the borders, by the way, before I move on? Clear? Okay, perfect. So now we're going to talk about the contents that are present in the middle ear. Someone's asking me, so the tympanic plexus is in the middle ear? Yes, it is in the middle ear. So the glossopharyngeal nerve gives a branch that is called the tympanic branch, enters the middle ear, and forms the tympanic plexus inside the middle ear. It is present in the middle ear. Okay, of course. So now we're going to talk about the contents. Now, when it comes to the contents, we have either bones or muscles that are present. So, sorry, both of them, obviously. I mean, when it comes to talking about them, we're going to start explaining the bones. So the bones that are present in the middle ear are the smallest bone in the entire body. SubhanAllah, it's crazy how important they are, and they're very, very small. Like if you look at any picture comparing them to your hand size, they're insanely small. And we have three of them. We have the malleus. Remember the handle of the malleus that we said we can see it while visualizing the tympanic membrane? Well, this is the malleus, and that's the handle of the malleus that we can see. So this is the first ossicle. We refer to them as ossicles. The second bone is called the incus, and it articulates with the third bone, or the third ossicle, which is the stapes. Now, the major function of these ossicles is to amplify the sound. So whatever sound we hear, in order for it to hear it properly, in order for it for us to overcome any resistance that is present These bones are present there to amplify the sound, so we hear every sound that's around us. So it's important for amplification. The opposite is true for the muscles. So we have two muscles that are present. We have the first muscle, which is the stapedius, which is this very tiny muscle. And from the name stapedius, it's attached to the stapes, which is this bone here. And an important thing about the bone stapes, let's go back to the bone, it's very important in transmission of the sound, and the way it does so is that it keeps vibrating against a structure that is called an oval window. If you don't get it now, it's okay. We're going to talk about it later again, but I just want you to understand the idea. So this footplate of stapes, or the stapes, it keeps vibrating and kind of tapping against the oval window. And this tapping is what causes transmission of sounds. Now, the stapedius muscle, what it does is that it pulls against the stapes, so it doesn't allow it to keep tapping so loudly, so it kind of decreases the transmission of the sound. We call it dampening the sound, meaning it decreases the sound that is being transmitted. Now, this stapedius muscle is innervated by a branch of the facial nerve. So you can guess if there is paralysis of the facial nerve, it can affect the stapedius muscle, leading to hyperacusis, meaning hearing very high sounds. Now, the other muscle that is present, which is this muscle here, is called the tensor tympani muscle. Now, the tensor tympani, as the name indicates, it tenses the tympanic membrane. Okay, I'll just kind of briefly touch upon the physiology so you understand the function. The way the sound is transmitted, we mentioned that it's going to be tunneled through the external ear, and then it's going to go to the external auditory meatus, and then what's after the external auditory meatus is our tympanic membrane. Now, these sound waves that are coming, they will cause vibration of the tympanic membrane. Basically, majority of the things we're going to discuss now is purely based on vibration. Now, as the tympanic membrane vibrates, we see here that the malleus is attached to it. This will cause further vibration in the malleus, and then followed by vibration in the incus, and then vibration in the stapes. And then I just explained how the stapes starts tapping against the oval window. So they're all kind of vibrating. Now we see the tensor tympani here. It is attached to the malleus here, to the handle of malleus. And the way it works is that it tenses the tympanic membrane by kind of contracting and pulling on the malleus. So the tympanic membrane doesn't vibrate as much, so the transmission of the sound decreases. These are all protective mechanisms for us, so we don't hear very loud sounds that can potentially damage our ear. So the ossicles are mainly for amplification, so we can hear the sounds properly, whereas the muscles, which are stapedius and tensor tympani, they're mainly for dampening the sound or decreasing it by either tensioning the tympanic membrane or kind of pulling on the footplate of stapes, decreasing its vibration. And the tensor tympani is innervated by the mandibular branch of the trigeminal nerve. And this is important, again, because they might tell you, a scenario that shows you, the facial nerve is paralyzed, or another scenario that shows you, the mandibular branch of the trigeminal is paralyzed, and they ask you what is the cause of hyperacusis. So you need to know which muscle is going to be paralyzed in this case. Okay, is that clear so far before I move further into the middle ear? Clear? Okay, perfect. Okay, so now we're just going to see the pathway spread of infection because, sorry, someone is asking a question. Tensor tympani paralysis can also cause hyperacusis. Yes, it can also cause hyperacusis because they're both responsible for dampening the sounds. And the majority of the cases they're going to ask you, it's going to be stapedius. Tensor tympani is mainly responsible for like chewing. So when the person's chewing, they would start to hear very loud sounds. It's going to be the case of tensor tympani. And it should make sense to you because if you study the mastication lecture, majority of the muscles that are involved in mastication are innervated by the mandibular branch of the trigeminal nerve. I don't know if that makes sense, but I kind of had this connection ever since I was a second year. Okay. So now we're going to see the pathway for the spread of infections in the middle ear. So if the person has otitis media, we're just going to skip the strictures. How does the infection spread? So here we have the external acoustic meatus, then we have here the tympanic membrane. Oh, by the way, another name for the middle ear is the tympanic cavity. So whenever you see any textbook or any question saying the tympanic cavity, it's like saying the middle ear. Then inside the middle ear, we discuss the different bones or ossicles that are present, malleus, incus, and stapes. Then we have the stapedius muscle, and then the tensor tympani. And then tegment tympani is the roof of the middle ear cavity. Then we have the eustachian tube. So a case of pharyngitis, or we said upper respiratory tract infection, it can spread through the eustachian tube to the middle ear, causing otitis media. So this is how it starts. So the patient can start having pharyngitis or any upper respiratory tract infection. It spreads through the eustachian tube all the way to the middle ear, causing otitis media. Now, and then because of otitis media, the tympanic membrane starts bulging, and one complication can be the tympanic membrane can rupture. Another possible complication is that it can spread through the roof or the tegment tympani, causing epidural abscess or meningitis, or can complicate into brain abscess or lateral or sigmoid sinus thrombosis. Or it can sometimes even affect the facial nerve, because we mentioned in no branches of the facial nerve pass through the middle ear. So it can affect the facial nerve, causing facial nerve palsy, but that's generally a very rare complication. Now, the mastoid air cells that are present behind our ear, it can also cause mastoiditis if the infection spreads there. So this is how the infection can spread. Now, acute otitis media, this is generally exactly what I talked about, just in words. And I don't generally use the slides of the doctors in my past sessions, but the slides of Dr. Yaqeen are actually really good. If you, when you're studying the lecture, try to like play them, like see the transition of the slides. It can really help you understand it. Okay, so now we're gonna move to the inner ear. Now, if you've noticed, the external and the middle ear were mainly responsible for the transmission of the sound. So there was not really any hearing that is present. We didn't see the process of us receiving the sound or understanding what we're hearing, because this is the major function of the inner ear. So the inner ear is the one where the hearing exactly happens. External and middle ear are mainly just transmitting or funneling the sounds into the inner ear. So we have the cochlea, which is this structure here, that is circular or snail-like structure, this one here. The cochlea was the one that is bulging on the medial wall of the middle ear, and it formed the promontory. So this is the cochlea. And then we have the vestibule, which is this structure here. And then we have the semicircular canals. We have 3 of them. Now, when it comes to the cochlea, it's mainly responsible for hearing. However, the vestibule and the semicircular canal are mainly responsible for balance and equilibrium. Now we're going to talk about some general concepts about the sounds. So we know, or I don't know if you know, a sound, it results from compression and refraction of air molecules. So the way we hear or the way we receive any sound, it's due to like kind of changes in air molecules, changing in the pressure of the air molecules. This will cause compression and refraction, and this is how we receive the sound. So there are 2 characteristics of the sounds that we kind of divide the sounds according to. So the first one would be the frequency. Now, if you look at this picture, this will kind of make you understand what a frequency is. It shows you the difference between a low frequency and a high frequency. So a low frequency sound is when we have very little number of like sound waves present at a specific period of time. So let's say this is both of these parts, low frequency and high frequency, are in 1 second. So in 1 second, we had just like, for example, 3. This is unrealistic numbers just for the sake of understanding. So we had 3. This shows us that it's a low frequency. However, here we had around 7 or 8. This shows that it's a high frequency sound. So how many sound waves are present in a specific period of time is what we refer to as a frequency. Now, the frequency determines the pitch of the sound. The pitch of the sound is basically when someone is playing piano, and in the piano we have different notes. The way you can tell that one note is different than the other is that's because they have different pitches. So that's how this is why the pitches are important. Another important thing, it's very important, it came in our final exam, is that females generally have a higher pitch, females sound, than males. So now that I'm speaking, my voice is generally classified as a high frequency or a high pitch sound. Whereas if it was a male explaining, it would be classified kind of a lower pitch sound. That's important, and it came in our final. Another important characteristic of the sound is the intensity. Now, the intensity is measured in decibels, and it's directly related to the amplitude or the loudness of the voice. So when I raise my voice, it means my voice has a high amplitude. That is a loud or highly intense voice is what we call the amplitude. However, the quieter sounds, if I start whispering, this will show that we have a very little amplitude, and it's where the quiet sounds are. So that's just general concepts or features of sounds that we must understand before dipping into furthermore. So now we're going to start talking about the cochlea. Remember the snail-like structure. We're going to talk about the anatomy. So here I want you to focus with me, because if you understand the anatomy, it's going to make it very easy. The cochlea is probably one of the toughest. It's not hard at all, but it's kind of tough to understand initially. Once you understand it, the physiology becomes easier and the rest of the anatomy is really easy. So let's imagine that we took this cochlea and we kind of cut it to see what's inside the snail-like structure, okay? So starting here, you see this cone-like structure that I'm highlighting? This is a bone that is called the modulus. So the cochlea, I want you to imagine it that is rotating in like kind of a snail look around this bone that is called the modulus. Now the modulus is cone-shaped, so it has a very wide base, narrow apex, and so on. One important structure is called the spiral lamina. The spiral lamina is kind of a very thin, bony protrusion from the modulus. So the modulus is a bone, and it gives us a very small bony protrusion that is called the spiral lamina, which is this structure here. So why is that important? Why are we studying it? Now, out of the spiral lamina, we have very important structures arising. From the spiral lamina, we're going to have two membranes arising. So the source of the membranes, or where they start originating, is going to be this bone, the spiral lamina that is coming from the modulus. So we have two membranes arising, and these two membranes will sort of divide the cochlea into three major parts. We're going to see them in the next slide. Okay, so imagine that I took this section here, I cut it, and we're looking at it at this picture, okay? Before I move on, I want you to understand it. Did you understand what we're doing so far? I don't want to go further without you getting the major concept here. So do you understand the structure initially? Okay, perfect. So now we cut this section and we're gonna zoom in. So see the membranes that I just drew? These are these two membranes. And these two membranes, because we have two, it's separating it into three parts. So this is the first part, this is the second part, and this is the third part. So the first part is called the scala vestibuli. The second part is the scala media, because it's in the middle, and the lower part is called the scala tympani. Now, the scala vestibuli and the scala tympani, they both have a fluid that is called a perilymph. Now, what is the perilymph? A perilymph is a fluid that is high in sodium, and it's very rich in sodium, and it doesn't have much of potassium. That's for scala vestibuli and scala tympani. However, scala media is rich in fluid that is called endolymph. So we were in perilymph, now we're in endolymph. Endolymph is the exact opposite. It's very rich in potassium and it's very low in sodium. We're going to see the significance of this when it comes to the physiology, but now just know this and keep it in your mind and know whenever we say endolymph, meaning it's rich in potassium, whenever we say perilymph, meaning it is rich in sodium and low in potassium. Okay, now let's talk about the membranes. So the first membrane here in blue is called the Reissner's membrane or the vestibular membrane. It can be used in both names. For simplicity, I'll just refer to it as vestibular membrane because it should remind you of scala vestibuli. Just know that it has another name, which is Reissner's membrane. Now, this vestibular membrane, it's what's separating scala vestibuli from scala media. So far, it's easy and self-explanatory. Now, the other membrane, which is very important, like I want you to know this membrane by heart and all the structures that are inside that I'll be explaining later, which is the basilar membrane. Now, this basilar membrane, it's what's separating the scala media from the scala tympani. And if we look at the basilar membrane, we're going to see different structures that I'll talk about later, but just know, the basilar membrane contains structures that are very important when it comes to hearing. Now, let's look at this picture here. It's kind of, we took this and we cut it again, like this whole structure here, we're just cutting it and we're going to see what's inside further. So this is our modulus. We're going to see these scala vestibuli, scala tympani, scala media, they're also present here. And we see that there's the cochlear nerve present. Now, the cochlear nerve is the nerve that is mainly responsible for hearing. It's part of the vestibulocochlear nerve. We're going to talk about it later, furthermore, when we go to the physiology part. Just know that the cochlear nerve is kind of connected to the to the cochlea, which makes sense. Cochlear nerve, cochlea, but it's very important in hearing. Just know that it's present here. And we're going to talk about a very important structure in the basilar membrane, which is the organ of Corti. So we took this and we further zoomed in. So the way I organize the slides is that we go from the outermost to the inner. So we were here, then we zoomed further more, and now we're taking this and we're zooming further. So we're here, okay? So we have the scala vestibuli, Reissner's membrane, and then here we have the scala media, basilar membrane, and then the scala tympani. Now we're going to focus on the scala media because it's the most important, and we said that it has endolymph. Now, the reason the scala media has endolymph, or the source of the very rich potassium in this fluid, is this layer that is called stria vascularis. So stria vascularis is kind of a layer of cells that are constantly, at a constant rate, releasing potassium into the scala media. That's why the endolymph at the level of scala media is very rich in potassium, in contrast to scala vestibuli and scala tympani, where we don't see the striae vascularis, okay? Now, a very important structure that is present on the basilar membrane, so this is our basilar membrane here. An important structure here is called the organ of Corti. Now, the organ of Corti is mainly formed of two things. We have the hair cells that are present, whether it's inner hair cells, these are the inner hair cells, or outer hair cells. And on top of the hair cells, we have this membrane that is made of glycoprotein that is called the tectorial membrane. So collectively, the hair cells, inner and outer hair cells, and the tectorial membrane is what we refer to as the organ of Corti. Now, the organ of Corti is the sensory organ for hearing, and it's the sensory organ that is present in the cochlea, because we're going to see multiple sensory organs present throughout the different sections of the inner ear. But when it comes to the cochlea, the sensory organ is the organ of Corti. And this is a very common MCQ that comes almost in every batch, okay? So this organ of Corti is the sensory organ of hearing. So when it comes to the physiology of hearing, it's going to be literally focused on this tiny structure here. Now, this picture doesn't show it, but I want you to imagine it. It's present throughout the whole length of the scala media. Now, another name for the scala media is called the cochlear duct. I don't want to overwhelm you with names. That's why I'm kind of introducing them slowly. So scala vestibuli just doesn't have a name. When we say scala vestibuli, it's scala vestibuli. Scala media can also be referred to as the cochlear duct. So within the cochlear duct or within the scala media, we have this organ of Corti that is the sensory organ of hearing, and it's made of hair cells, inner and outer, and the tectorial membrane. Now we again zoomed into the organ of Corti. So we're taking this section and we're zooming in. So we keep zooming in to understand the structures. So the inner hair cells, a very important thing about them is that they're made of one layer of hair cells. So we only have like one cell. There are no multiple layers of cells. However, the outer hair cells, we have different layers. 3 to 4 layers of hair cells form the outer hair cells. And this is the tectorial membrane that is on top of them, and that's the organ of Corti. Now, if you look at the picture, you're going to notice that the cochlear nerve is mainly present in the inner hair cells, or mainly kind of synapsing with the inner hair cells. And this is very important because the inner hair cells are the cells that are mainly responsible for hearing. So 90 to 95% of our hearing is through the inner hair cells, whereas the outer hair cells are mainly responsible for modulation of the sound and protection of the inner hair cells. So they're kind of responsible for protecting our inner hair cells. But the hearing itself comes from the inner hair cells. Now, if we take the hair cells and we zoom further deeper, this is what it looks like. Okay, so we kept zooming. We reached to the point where we reach the hair cell and we're zooming in. The hair cell has stereocilia on top of it, which are just cilia عادي. And then we have the kinocilium, which is one, which is the tallest out of all the stereocilia. And this is important to the physiology, so keep it in mind. So we have multiple stereocilia and one very tall kinocilium in every single hair cell. And these hair cells and like the tectorial membrane and the organ of Corti, they're present throughout the whole length of the cochlear duct or the scala media. So the picture again doesn't show it, but I want you to imagine they're present throughout the whole length of the scala media. Okay. So here we're going to look at the cochlea. Now we zoomed out to see the cochlea with the other parts of the ear, so the middle ear and like the eardrum. An important thing here is this structure that we call the oval window. Now this, if you look here, this is the middle ear. We have the malleus, incus, and stapes. So this is our stapes. And we said the foot plate of stapes keeps tapping on the oval window. So this is where it's tapping. I'll just change the color. Okay. So this is where it's tapping. Now, because it's tapping on the oval window, we said we started with vibration in the eardrum, then vibrations in the ossicles, and then tapping on the oval window. Now, the oval window is mainly present in these scala vestibuli. Now, I'll just, again, orient you. This is the scala vestibuli. This is the Reisner's membrane here. This is the cochlear duct or the scala media. And this is our basilar membrane, and that is the scala tympani. Okay? So the foot plate of stapes tapping on the oval window is mainly present in the scala vestibuli. So it keeps tapping. Now, because it's tapping, and we said the scala vestibuli is rich of fluid, there's the perilymph. Now the perilymph would start to vibrate because it's a fluid. And now basically the point where the mechanical vibration, which is in the bone, turns into fluid vibration is at this point, where the foot plate of stapes taps over the oval window. This will cause vibration of the perilymph that is present in these scala vestibuli. So here we're in scala vestibuli. Now the fluid or the perilimb starts vibrating, the vibration will go all the way. It reaches this point. Now at this point here, we see that the scala vestibuli kind of mixes with the scala tympani, because this is our scala tympani. So the perilimb of both meet at this area here. This area is what we call the helicotrema. It was present earlier, but I didn't think it was clear. I think this is the best place to explain it. The point where the scala vestibuli and scala media join together is what we refer to as the helicotrema. Okay, now that's just for anatomy. Let's go back to the fluid that was vibrating. So the perilymph is vibrating. This, because it's vibrating, this will cause then vibration through throughout the whole perilymph until the vibration reaches the basilar membrane. Now, once the vibration reaches the basilar membrane, this is where basically the hearing is starting. Okay, so now we have a question before we go into the physiology, because now we're almost done with the anatomy of the cochlea. We're gonna go to the physiology. I'm not gonna explain the anatomy of the semicircular canal and the vestibule. I'll explain it at the end of the lecture. Now we're gonna focus on the cochlea and the hearing. So can someone answer this question? It's very easy. Okay, so you all answered C, and the answer is correct. The part of the cochlea that contains the organ of Corti is the scala media, which is the cochlear duct, and that's where the endolymph is present. Okay, perfect. So now we're going to talk about hearing. So how does the person hear, or how are the sound perceived? Okay. So hearing, sound, we discussed that it's like when the air molecules are refracting and compressing and so on. This is how we start hearing. So it's like a pressure wave that is coming. That's basically what a sound is. Now the human ear can only hear sounds when it comes to frequency that are from 20 to 20,000 hertz. Anything below 20 or anything above 20,000 hertz in terms of frequency, we don't hear it. Someone's asking me if we can have 5 minutes break for prayer. Yes, sure. We'll have 5 minutes break, then I'll start with the physiology of hearing. No problem. Okay, sure. So we were talking about the physiology of hearing, and we said that the sound is mainly due to pressure waves that are present in the air molecules that are received by our ears. And we said that the human ear can only hear the sound waves between frequency 20 to 20,000 hertz. Anything beyond 20,000 or below 20, the human ear cannot hear that. And the safest amplitude of the sounds, if it's higher, it would be dangerous for our ear. So the safest would be from 1 to 80 decibels. Anything higher than that is considered something that can potentially damage our ear, because very loud sounds can damage our ear. Okay. So the amplitude, as we said, is the total change in pressure. Now the peak sensitivity, or the best frequencies for the human ear to hear, is between 1,000 and 4,000 hertz. Whenever we say sensitivity, it's the ability to hear even the very quiet sounds, and the acuity, it's the ability to discriminate the sounds. So the peak of both sensitivity and the acuity is between 1,000 and 4,000 hertz. And subhanAllah, a speech, or when someone is talking to you, it's usually between 250 to 4,000 hertz. So this is where our peak sensitivity and acuity is. It's at this just regular speech level. Now the sound transduction, we turn the air waves into mechanical vibration, and then fluid vibration, then chemical signals, finally action potential, which is what we discussed. So we had the air waves in the ear, they come to our ear, then there would be mechanical vibration from the tympanic membrane, the ossicles, and then footplate of stapes. This mechanical vibration gets converted into fluid, because we said the footplate of stapes starts tapping over the oval window. The perilymph vibrates, and then the endolymph vibrates, and then there'd be chemical signals and action potential. That's the physiology that we'll talk about it, inshallah. And then the initial processing of the pitch, loudness, and duration of sound takes place in the cochlea. We discussed that also. And localization of the sound, meaning whenever you hear a sound and you want to know what's the source of the sound, it occurs at higher CNS levels. It occurs at a structure in our brain that is called the superior olivary nucleus. So it does not occur at the ear level. So if it's just the function of the ear itself cannot tell you what's the location of the sound, or what's the source of the sound does not happen at the level of the ear. It happens at higher levels in the CNS, mainly in the superior olivary nucleus. So that's also very important information, can come as MCQs. Okay. So we're just going to revise the anatomy of the ear before we go into the physiology. So we had the external acoustic meatus. We said the sound waves come here, vibration of the tympanic membrane, and then we'd have the middle ear, also known as the tympanic cavity. We have the ossicles, we have the malleus, the incus, and stapes. Now the stapes start stabbing over the oval window. The tegment tympani is the roof. We know that. Okay, we already discussed it. And I just wanted you to see kind of how the stapes taps over the oval window, and now we'd have like vibration in the perilymph and so on, and reaches the cochlea, which is how the hearing is felt. So, and we have the scala vestibuli, scala media, and scala tympani. And then we had the cochlear nerve, and the cochlear branch goes and the cochlear branch of the vestibular cochlear nerve goes from the cochlea and joins the vestibular branch, which we're going to talk about later. Okay. Now, the role of the middle ear in hearing. Remember, we already talked about the ossicles and how they amplify the sounds. So that's their main function. And that's a mechanism that is called impedance matching. So through the ossicles, there is a very important function, which is the impedance matching. Now, what does the word impedance mean? Impedance is basically any resistance or obstacle in the pathway of the sound. So while the sound is passing, there are multiple obstacles that it's facing. One of which that are faced in the ear is the fluid, because the resistance in the fluid is much higher than the resistance in the ear. Let me just spell the chart. Okay. So we know that the resistance in fluid is much higher than the resistance in the ear. And that's important because if you want to hear a sound as accurately as you're hearing it from the environment, you need to increase the pressure in order for you to overcome the resistance that is present in our ear. Because we know inside the cochlea, it's all fluid. We have perilymph and endolymph, which means there is very high resistance. So the way our body overcomes this high resistance in the inner ear or the fluid is by a mechanism that is called impedance matching, which is basically through the ossicles. So, okay, that's something I've already mentioned. So the way it does so is by two mechanisms. So the ossicles, they work in a chain. So like they increase the force movement in around 1.3 times. And another very important information is that the tympanic membrane is 17 times larger than these tiny ossicles. So when you're exerting a pressure over a very tiny structure, as compared to a very large structure, the pressure would be way higher, in the smaller structure. So say, for example, you have a very large surface area and a very small surface area, and you're applying the same amount of pressure on both, the pressure would be way higher in the small surface area because there is no surface area for the pressure to be lost. So now because the ossicles are 17 times smaller than the tympanic membrane, so this will increase the pressure 17 times. And we already had the lever movements acting in a chain, like increasing it by 1.3 times. So the total increase in sound waves would be 22 times in the ossicles. And that's what we refer to as the impedance matching. This is all to overcome the resistance that is present in the fluid in the inner ear. Does that make sense? Like, do you understand it so far? What's impedance matching? Yep, okay, perfect. Now we're going to move to the other important function of the middle ear, which is what we call the attenuation reflex. Now, this is the exact opposite of the impedance matching. Attenuation reflex is activated whenever we hear very loud sounds. So imagine that you're in a party and there are very loud music there that can potentially harm your ears. Like say, for example, you're sitting next to the radio that's playing very loudly or whatever. So there is a very high sound. So our bodies, subhanAllah, has a way of, to deal with that, which is by what we call an attenuation reflex. So the way it works is by the muscles that we just talked about, the tensor tympani and stapedius. So we said the tensor tympani, it pulls the handle of the malleus. This tenses the tympanic membrane, so it reduces the vibrations, and the vibrations are the main way of how the sounds are transmitted, so it reduces the sound transmission. And the other way is the stapedius, that's pulling in the footplate of stapes, so reducing the tapping over the oval window. There's a small delay by which this attenuation reflex activated, which is 40 to 80 milliseconds, and generally it will decrease the intensity of the sounds by 30 to 40 decibels. Okay, so far there isn't much to explain in these. So yes, now we're going to talk again about the organ of Corti. So we're back to it. So this is our organ of Corti. These are the hair cells, and that is the tectorial membrane. And we said it's present in these scala media here. Okay. So the hair cells are divided into inner hair cells that is made of single row and outer hair cells. Now these hair cells, they were synapsed with the spiral ganglion of Corti. Okay, so if we go back, I'll actually go back to show you in one of the slides. Okay. Okay, here. Oh, it stopped sharing. 1 second. Okay, perfect. So if you go back here, we're going to see that there is a spiral ganglion that is present near each kind of organ of corti. So the way is the spiral ganglion, remember when we were talking in neuro, like about generally the spinal tracts, and we had the first order neurons in the dorsal root ganglia, which was a pseudounipolar neuron. This is a very similar concept. So we have this, for example, this is our spiral ganglion, and this is our hair cell. The way it works is that the spiral ganglion has a peripheral process because it's a pseudounipolar neuron, and a central process. Now, the peripheral process will go to the inner hair cells, and the central process will go to the CNS, to the central nervous system. This central process is the cochlear branch of the vestibulocochlear nerve. So this is our central process, and here this is the spiral ganglion. I want you to imagine that there's peripheral processes that are going to the inner hair cells and the organ of corti. So this is how it works. Now I just explained it so that you understand the concept. Let's go back here. Okay. I don't know why they're saved, but okay. So going back to the receptors in the organ of corti, we talked about them, the inner and the outer hair cells, and we said that 90% of the auditory nerve fibers are innervated by the inner hair cells. So the inner hair cells are the ones that are mainly responsible for our hearing, whereas the outer hair cells are mainly to protect the inner hair cells. So they modulate the sounds and they control the sensitivity. We're going to discuss it even more. So let's see what we've discussed so far. So going from the external ear, passes through the external auditory meatus, vibration tympanic membrane. Now the ossicles would vibrate. Footplate of stapes starts tapping against the oval window. This will cause vibration of the perilymph. Eventually, the vibration reaches the basilar membrane, where the hair cells are present. So going back to the hair cells. Okay, so we had the stereocilia and the kynocelia that we talked about them. Now, a very important connection between the stereocilia, different types of stereocilia, and the kynocelia is these tiny lines, tiny black lines between them that we call the teplinks. Now, these teplinks that are connecting the different stereocilia and the kynocelia, they contain potassium channels. Now, this is very important because this is like how the hearing is actually received, like at this point. So we're talking how we have endolymph and the scala media. So let's draw it. So we have, this is our scala vestibuli. This is our scala media, and this is our scala tympani. This is our basilar membrane. Here we have the organ of corti, we have the hair cells, and we have the tectorial membrane. Okay, so we're going to zoom in. So this is the hair cell. Here we have the endolymph. Let me just draw the endolymph. So let's imagine that this is the endolymph that is going and moving. Whenever the endolymph starts vibrating, because we have already vibration in the ossicles and then perilymph, and it reaches the endolymph. Now the endolymph, because it's vibrating, it's kind of going to move in between the tectorial membrane and the inner hair cells in the organ of corti. This endolymph movement between them will cause these stereocilia to bend. Now there are two possibilities for the bending. The bending would either go towards the direction of the kynocelia, which is this case. So you see the stereocilia are bending towards the tallest out of all of them, or it's going to go against the direction of the kynocelia, which is this case. Now, if it's in the first case, where the stereocilia are bending towards the kynocelia, this will open the potassium channels that are present in these step links, and potassium starts to rush inside the hair cell. So we'd have potassium going inside. And remember, we're in scala media. This is why we have lots of potassium, because we said we have stria vascularis, we have endolymph, lots of potassium. This is where potassium comes to play. The endolymph moves between the tectorial membrane and the inner hair cells. It bends the stereocilia of the hair cells. The bending, if the bending takes place towards or in the direction of the kynocelia, the potassium channels that are present in these step links would open, and potassium starts to rush in. Now, these potassium ions would depolarize the cell. And because it is depolarizing the cell, It's going to open the voltage-gated calcium channels. So we also have voltage-gated calcium channels present. When potassium enters, it depolarizes, reaches the threshold, calcium enters inside the cell. And once calcium enters, and we know calcium is a very important for ion for like releasing whatever neurotransmitter we have. Calcium causes fusion of neurotransmitters that are already present inside the inner hair cells into the cochlear nerve. Now, this neurotransmitter here that is being released is glutamate. Okay? We're going to repeat it again, but no, this is how we did here. Like, it's as simple as that. So the endolymph that is very rich in potassium is vibrating. We've reached this point. We discussed all that, and we reached the point where there's vibration of the basilar membrane and the endolymph and so on. Now, the vibration of the endolymph will cause the endolymph to move. It causes bending of the stereocilia. If the bending of the stereocilia is towards the kinocilium, this will open the potassium channels that are present in these step links. Potassium rushes inside the cell, depolarizes the cell, opens the voltage-gated calcium channels. Calcium enters the cell. Calcium causes fusion of these glutamate, the neurotransmitter glutamate, and then it's going to be released into the cochlear nerve. And now we have an action potential, and now we start to hear. This is just words of what I said. Is that clear? And did you understand this part? I just want to see where the chat is because I can't really see your. Okay. Yes. Okay. Perfect. Does anyone have any question? Because if you understand this part, it makes it easier for like the rest of the physiology. If not, Adi, I can re-explain. Okay, crystal clear, perfect. What does the protein bridge called, please? It's called TEP links. TEP links. Wait, I have it written somewhere. Okay, here. The TEP links. It's the connection between the stereocilia. Does anyone have any other question? Okay. Now we're going to talk about the other case, where the stereocilia bends against the direction of the kinocilium. Now, if the stereocilia bends against the direction, the TEP links will not be activated, the potassium channels won't open, and we will not have action potential or potassium entering inside the cell. So the key to hearing is that the stereocilia bends towards the kinocilium. If it bends on the opposite direction, then we don't have everything that we just discussed. And let me just check if I repeat it, if I forgot anything. Yes, okay. Now the cochlear nerve here, it's an afferent neuron. So we have like these glutamate or neurotransmitter glutamate going to the afferent neuron, so it reaches the brain, so we perceive our hearing. However, when it comes to the outer hair cells, they're generally connected to an efferent neurons that release acetylcholine, not glutamate, and they contain Preston molecules in their lateral wall. Okay, so I'm just gonna draw it because I don't think there is a picture that shows, no, there is not. So I'll just draw it. So we said we have the inner and the outer hair cells, so we're here. Now we're going to talk about the outer hair cells. The outer hair cells are connected to these efferent axons and they release acetylcholine. Now, why is that important? We said that the outer hair cells are mainly protecting the inner hair cells and they're modulating the sound. But how do they do that? Now, in between the outer hair cells, there are like small protein molecules that are called Preston molecules. Imagine that these protein molecules that I'm drawing is what we call Preston molecules. Now, the function of these Preston molecules is that whenever we have a very loud sound that is being heard, to protect us, these Preston molecules elongate. Okay? So these molecules would just elongate and the outer hair cells overall would elongate. This pushes the tictorial membrane away from the inner hair cells. Now, this is important because when it does so, when it pushes it away, because we said the vibration of the basilar membrane is the basis. The basal membrane vibration won't reach the tictorial membrane. There won't be bending of the stereocilia. And now this will kind of dampen the sound. So this is how it protects the inner hair cells, because if we have a very loud sound and the inner hair cell is receiving the sound like fully, this can severely damage the inner hair cells. This is why loud sounds are very dangerous, because they can damage the inner hair cells in particular. So the outer hair cells are, subhanAllah, designed in a way that they have these Preston molecules that would elongate it. They would kind of separate the organ of corti. They would have the tictorial membrane on top and the basilar membrane down. And because they're separated, because of the elongation, you won't have the action potentials generated, or at least they would be decreased. Does that make sense? I know it's not an easy concept to understand, but يعني just know the crystal molecules would elongate it, and خلاص, now this is a protective mechanism, and this is how it modulates sound. Okay, alhamdulillah, you all understood? Okay, that's perfect. Okay. Now the sensory coding. What it means is basically how do we differentiate between different sounds when it comes to frequency and amplitude. So let's start with frequency. Remember, let's go back to the example, a female and a male. We said females have a very high pitch sound, yani they have a higher frequency. Or any sound in the world that has a high frequency, it's going to be heard at the base of the basilar membrane. So this is our basilar membrane that we talked about. We have a base and we have an apex. And remember, the base is like, it's near the oval window, near the stapes. So this is where the base is. Whereas the apex is further away. Now in the base, the basilar membrane is very stiff and it's very narrow. And this is where the high-pitched sounds or higher frequency sounds are heard, at the base. Whereas at the apex, it's generally wider and loose. It's more flexible at the distal end. And this is where the low frequencies are heard. So when we say base, it is for high frequencies. And when we say apex, it is for low frequency. Okay? I want you to remember this really well because it comes a lot in MCQs, like almost every batch. So we said high frequencies are heard at the base and the low frequencies are heard at the apex. So this is how we code the pitch or the frequency of the sound, or this is how the brain translates it. Now the amplitude is different. The amplitude is kind of the extent or the degree of like the action potentials that are being activated. So we said we have hair cells throughout the whole length of the cochlear duct or the scalomedia. We discussed that. So the amount of hair cells that, let's say we just activated the hair cells up until here. So that's considered kind of a low amplitude. However, if you activate the hair cells all the way, that is a high amplitude sound. So the amplitude is just the degree of displacement and or the amount of action potentials that are generated, or the extent of activation of the inner hair cells. Like how much of the basilar membrane is basically activated. Does that make sense? That's the difference between frequency and amplitude. That's how the body perceives it. Okay, perfect. Now we're going to move to the hearing pathway. Now, know one thing, that the pathways in head and neck are generally not as important as they were in neuro. If they come, they're just going to ask you which structure is involved or which structure is present. So you don't need to know them in details. Just know the general concepts and know the different structures that are involved. That's it. You don't need to know the details that or the level of details you were studying back in neuro. However, I'm just going to explain it again. So remember that we talked about the spiral ganglion that we talked about. So we had the peripheral process that is going to the hair cells, and then we had a spiral ganglion, and then the central process that is going to the CNS. Okay, so we're going to go back to that example. So the spiral ganglion in the modulus that we just saw, the peripheral process, and the central process forming the cochlear part of the vestibule cochlear nerve. That's all information that I just explained before. Okay, yeah. So we have the cochlear nuclei. So we have ventral and dorsal cochlear nuclei. I'm actually going to draw it here. So let's imagine that this is our brainstem. Okay. And at the junction between the pons and the medulla, we have the cochlear nuclei, where the central processes are going. So the cochlear nuclei, we have a dorsal group, and we have a ventral group. Let me color code it, actually. Okay. So this orange line is separating between the dorsal group in D and the ventral group in V. So these two are the ventral group. Now the ventral group is further subdivided into anterior ventral group and posterior ventral group. Okay, these are all the cochlear nuclei that are present in the brainstem. So we have ventral, anterior, ventral, posterior, and we have the dorsal group. So far, there isn't much that we're talking about in the pathways. We're just understanding what are the structures that are present at the level of the brainstem before diving into the pathway. I don't want to forget anything. That's why I'll keep referring to the slide. Okay. So let's start what happens when it comes to the dorsal group. So let's start with the dorsal group. So let's imagine this is our spiral ganglion. We had the peripheral process coming from the inner hair cells and then the central process, and it goes to the dorsal group of the vestibular nuclei. So it's going to form a structure. Let's color it. Wait, I can't see the colors. Okay, let's choose the color blue. It's going to form a structure here, this one, which is known as the dorsal acoustic striae. Okay, now this dorsal acoustic striae would cross the midline, so it will go to the contralateral side, and it's going to synapse with the lateral lemniscus nucleus. Okay, so we have another nucleus that is called the lateral lemniscus in the color green. So we started with the cochlear nerve. It goes to the CNS. It's going to synapse with the dorsal. It synapses with all of them, but in cases of the dorsal, when it synapses with the dorsal vestibular nucleus, it's going to form the dorsal acoustic striae. It's going to synapse with the nucleus of the lateral lemniscus. And then as it synapses with the nucleus of lateral lemniscus, let's actually stop here and then see what happens with the other pathways. So let's just know, and it's going to form the dorsal acoustic striae, synapses with the nucleus of lateral lemniscus. Now the ventral posterior, okay, sorry, this should have been the posterior and this should have been the anterior. Now the ventral posterior nucleus has a very similar pathway. So again, the cochlear nerve would go, synapse with the ventral posterior nucleus, and then it's going to cross the midline and synapses with the nucleus of the lateral lemniscus. What's unique here is the ventral anterior group. So you're going to have the cochlear nerve. It will synapse with the ventral anterior group of the vestibular nucleus. And when it synapses, it's going to go to another nucleus that is called the superior olivary nucleus. Let's color it with this color. Okay. Let's imagine that we have two superior olivary nucleus. Now, when it synapses with the ventral anterior part, it's going to go and synapse with both superior olivary nuclei. So the one on the right and the left. See, when it comes to the dorsal group and the ventral posterior group, it crosses the midline and synapses with the medial lemniscus on the contralateral side, and nucleus of the the lateral lemniscus. However, when it comes to the ventral anterior group, it's going to synapse with the superior olivary nuclei on both sides. So bilateral supply to the superior olivary nucleus. So why is that significant? Remember when we talked about the superior olivary nucleus earlier, and I told you it's very important for the localization of the sound, so you know where the sound is coming from? So this is how. This is because the superior olivary nucleus is bilaterally supplied from each ear. So it makes sense that the superior olivary nucleus localizes the sounds because it's the one that is receiving the sounds from both sides. Does that make sense so far? Before I go further in the pathway. I know the pathway is complicated. Honestly, I hated it when I was a second year, but inshallah, I'll try to simplify it. Can you please repeat it? Sure, I'll repeat. Sure. Okay. I'll delete it and just draw it better. Okay. So we said we talked about it earlier, we had the spiral ganglion that is present in the ear. Okay. Now the spiral ganglion had a peripheral process that is going to the hair cells. So these are our hair cells that are present in the ear, and we had the peripheral process of the spiral ganglion. Now, in the spiral ganglion, we had a pseudounipolar neuron, meaning it has a peripheral process and a central process. So the peripheral process goes to the inner ear. This is the spiral ganglion. And the central process will go to the central nervous system. Now, the central process is actually forming the cochlear branch of the vestibulocochlear nerve. And then it's going to go to the brainstem or the central nervous system. So far, is it clear? Do you understand this part? How we form the cochlear nerve from the spiral ganglion? Yes, okay, so I understand this part. Now we're just going to draw different nuclei that are present in the brainstem that the cochlear nerve can possibly synapse with. So the first nuclei that we're going to talk about are the vestibular nuclei. Now the vestibular nuclei have a dorsal group, of the vestibular nuclei, and a ventral group. Now in red, I'm drawing the dorsal. Okay, I'll if you get lost, just see, I'll just write everything here in the same color. I'm sorry for how I'm writing it, just for the sake of understanding. So this is the dorsal group, and then we'd have the ventral group. Now the ventral group is further subdivided into ventral posterior and ventral anterior group. So in green here, this circle here, is the ventral posterior vestibular nucleus. Okay, let me just draw ventral posterior, okay? And then in blue, I'll have the ventral anterior vestibular nucleus. The cochlear nerve would synapse with all these three nuclei, but each one of them would have a different pathway or a different way. Vestibular nuclei or cochlear nuclei? No, they're okay. I'm sorry, they're the cochlear nuclei. Yes, you're correct. I'm sorry. Yes, they are the cochlear nuclei. Never mind. The ventroposterior part of the cochlear nucleus. Sorry, I just got lost because the vestibular nucleus is in the balance. Okay, it's the cochlear nucleus. Okay, so dorsal group of the cochlear nuclei, then the ventral posterior part of the cochlear nuclei, and the ventral anterior of the cochlear nucleus. It's written here. Okay, I'm sorry guys for the confusion. I was focusing on like ventral posterior. I didn't notice that I was saying vestibular. Okay, so it's the cochlear nucleus, okay? Do you all get it? It's the cochlear. Forget about the vestibular. We'll talk about it later. It's the cochlear nucleus, okay? Also, it's the cochlear nerve. Makes total sense for the cochlear nerve to synapse with the cochlear nucleus. It doesn't make any sense for it to be the vestibular. Okay, so now the cochlear nerve would synapse with the ventral posterior cochlear nucleus, ventral anterior cochlear nucleus, and the dorsal cochlear nucleus. Now, in case of synapsing with the dorsal cochlear nucleus, it will form like it synapses with the dorsal cochlear nucleus, and then it's going to cross the midline. While it's crossing the midline, it's going to form a structure that is called the dorsal acoustic striae. I'll just refer to it as the AS. Notice that it crossed the midline. So it went to the contralateral side. Now, when it goes to the contralateral side, it will synapse with another nucleus. Here, let's change the color actually. Let's say the color purple. This nucleus in purple is the nucleus of the lateral lemniscus. So we had the cochlear nerve enters. When it synapses with the dorsal cochlear nucleus, it's going to form the dorsal acoustic striae, which crosses the midline, synapses with the contralateral, because we have a nucleus in both sides. It will synapse with the contralateral, lateral lemniscus nucleus. Is that clear so far? Like, do you understand it so far? Because I'm going to stop here for the dorsal nucleus. Yes, okay. Now the exact same in case of ventral posterior nucleus. So the cochlear nerve would go, it will synapse with the ventral posterior cochlear nucleus, it crosses the midline and synapses with the nucleus of the lateral lemniscus. They both have crossed the midline. Now what's special or what's unique is the ventral anterior cochlear nucleus. You have different nuclei that are called the superior olivary nucleus. They're present kind of here at a lower level, actually. Don't focus on the anatomy in my drawing because it's not accurate at all. Just focus on the concept or the function. Now, the ventral anterior nucleus, the cochlear nerve would go, it will synapse with the ventral anterior cochlear nucleus. The ventral anterior cochlear nucleus would synapse with both sides of the superior olivary nucleus. So superior olivary nucleus. So bilateral supply of the superior olivary nucleus from both ears. Let's say, for example, here we're discussing the right ear. So it goes inside, supplies bilaterally, same for the left ear. Whatever you hear from the left ear would also go and supply the superior olivary nucleus bilaterally through the ventral anterior cochlear nucleus. Is that clear so far? Do you understand it so far? Yes, okay, perfect. Now, what's important to understand here is that the superior olivary nucleus, because it gets the bilateral supply, it's the organ or the part of our brain that is responsible for the localization of the sound. So whenever you hear a sound and you want to know like what's the source of the sound or from which direction is the sound coming, so that's the function of the superior olivary nucleus, because we mentioned earlier that does not occur at the level of the ear, at higher centers of the CNS, and that would be the superior olivary nucleus. Now, when we reach the nucleus of the lateral lemniscus, I'll use this drawing here because it's easier. Nucleus of lateral lemniscus on both sides, and we were talking about, for example, the right ear. Let's imagine we had the same for the left ear. Both information from both lateral nuclei, they would kind of cross over, and each lateral nucleus of lateral lemniscus would kind of convey the information to the other. This is a very weird way, subhanAllah, that it happens, but you really don't have to know the exact mechanism of how it does it, but just know the lateral lemniscus eventually would get the information from both sides because of the crossing that happens here. You see this blue line here? This is basically how the crossing between the two lateral lemniscus nuclei happens, so the information eventually would overlap. Now, these tracts, all of them. So let's imagine that this, the cochlear nerve forming the dorsal acoustic striae reaches the nucleus of the lateral lemniscus, going back to my drawing, and then going to the ventral posterior group, and then the ventral anterior group. All of them would eventually join together, and they will form a very thick tract. And remember, a tract is the collection of axons in the CNS. Now, this very thick tract is what we call the lateral lemniscus, and it will ascend. Now, this lateral meniscus eventually will pass through a structure that is called the inferior colliculus. And then it will go through the medial geniculate body and then to the cortex in order for us to hear. I know it's not an easy pathway at all. I can repeat if you want to, but this is how it works. Do you understand it for now? If not, عادي. Well, I'm willing to repeat it. It's fine. Because I struggled a lot as a second year studying it. I understand that it's a very tough pathway. Clear now? Okay. Does anyone want me to repeat? All good? Perfect. So the lateral meniscus collects all fibers from all nuclei. Yes, it does. It collects all fibers from all nuclei. Okay, of course. So let me just check if I have missed anything. So as we said, the superior alveolar nucleus is bilaterally supplied. Superior colliculus. Yes, and this is important because eventually we said that the fibers they will overlap, and then when it reaches to your brain, you basically have the information from both your ears because the fibers eventually would overlap. So why is that important? Because if يعني لا سمح الله someone has like a unilateral cochlear damage or deafness, they would still be able to hear fine because they have another ear because the information overlap eventually. What's coming from the right and the left eventually is overlapping. So if there is damage to one side, it does not affect on the level of the brain or the perception of the sound because eventually the supply is bilateral. Does that make sense? Unilateral cochlear damage would not result in like total loss of hearing from one side because خلاص, the information eventually overlaps. Okay, and I'm sorry for saying vestibular. Remember, it's a cochlear nucleus. It's cochlear nerve, so it makes sense for the cochlear nucleus. Vestibular is related to balance, not to hearing. Okay. Now we're going to talk about deafness. So deafness is inability to hear. Someone's asking me, what about localization? Is it going to be affected in deafness? No, it's not going to be affected because, as I mentioned, the superior alveolar nucleus is going to be bilaterally supplied from the beginning. So here, when the cochlear nerve synapses with the ventral anterior cochlear nucleus, it will go and supply the superior alveolar nucleus bilaterally, and the same is for the other ear. So if one ear is impaired, the other would still have like intact superior alveolar nucleus function. So no, the localization will not be affected. Okay, perfect. So the deafness is inability to hear, and we have two types of deafness. We have either conduction deafness or nerve deafness. The nerve deafness is also sometimes referred to as the sensory neural deafness, okay, because it's a sensation at the level of the neurons that is impaired. Okay. So when it comes to conduction deafness, as the name says, it's an impairment in the transmission of the sounds into the cochlea, because we know the cochlea is the major organ for hearing. Anything like before the cochlea is almost always a conduction deafness. So if there is a foreign body in the external ear that is blocking the sounds from going in, or for example, say there is otitis media that is damaging the tympanic membrane and the ossicles, because we know the major function of the tympanic membrane and the ossicles is to transmit the sound. They're not for hearing. So they're to conduct the sound. If they're impaired, then there will be conduction deafness. Okay? And then fixation of stapes into the oval window or osteosclerosis. So that's basically a disease where the oval window can no longer tab over, the stapes can no longer tab over the oval window. So basically there won't be any transmission of the sound because this like point of tapping of the foot plate of stapes against the oval window is crucial when it comes to hearing. So if there is any fixation, like because the stapes can no longer tap or osteosclerosis, this is also considered conduction deafness. Now, when it comes to nerve deafness or sensory neural damage, it can be either degenerative, basically when people get older, generally they would have degenerative process in almost everything in their body, whether their joints, whether like their hair cells. So degenerative process is considered a type of nerve or sensory neural deafness. Hereditary, if someone has like hereditary problems in their cochlea, it's not any properly developed or whatever, or some toxins such as quinine, measles, they all can cause sensory neural deafness. Acoustic trauma, because they can damage the inner hair cells, as I mentioned, meningitis, and acoustic neuroma, which is a tumor in the ear. Now, these are all considered nerve deafness because acoustic neuroma mainly would affect the nerves again. Now, there are different tests to differentiate between the types of deafness, and we're going to talk about them. So the first test is called Rainey's test. And this is mainly done to compare the air conduction and the bone conduction. Now, what does that even mean? So when we're hearing, we have two ways for us to hear. Either like the typical way now that I'm speaking to you and you guys are hearing me, that's through the air conduction. But we can also hear through the bone conduction because the bone can also conduct the signals and we can also hear it. And the major bone that we test the bone conduction in case of Rainey's test is going to be the mastoid bone, which is here. You see, like this guy here, we're examining the bone conduction in this picture because we're placing the tuning fork. I'm sure you've experienced the tuning fork in Pro. So now the tuning fork is vibrating. Now, as the tuning fork is vibrating, it can be placed like behind the ear on this, like on the mastoid. And when it's placed on the mastoid, let me just change the color. Okay. So when it's placed on the mastoid, there would be conduction through the bone to the ear. Now, in a normal person, this conduction would be, or this hearing through the bone conduction should always be like lower or not as high or not as loud as the air conduction. And the air conduction is simply when you place the tuning fork. Again, I'm sure you've heard it in Pro, like there's a sound when you place it, even when it's vibrating, and you place it near your ear so you can hear. That's an air conduction. So in a normal person, the air conduction should always be louder. Okay, so it's always louder as compared to the bone conduction. Now, if someone has a problem in their ear or they have a conduction deafness, the bone conduction would be higher than the air conduction. However, in case of nerve deafness, they will both be less perceived. Now, someone might ask me, why would the bone conduction be higher than the air conduction in case of Rainey's test? That's because in conduction deafness, now basically let's take any example of conduction deafness. Let's say the person has a foreign bodies in their ear or wax is just, there is too much wax production and wax has accumulated and so on. So this basically would block the background noise. So the bone conduction would now be even higher because there is no background noise to hear. The bone conduction would be heard because the nerves are fine, the person can hear. However, there is a conduction problem. The sounds are not being transmitted normally. But when it comes to the bone, now it's transmitted through a different mechanism. It's not through the hair cells and so on. Plus, I'm sorry, it is through the hair cells, but it doesn't go through the middle ear and like the external auditory meatus. No, it goes through like immediately from the bones, but it is through the hair cells. Anyway, point is, when you place it here and you have or the person has a conduction deafness, there is no background noise. You don't hear any noise like other than the bone conduction, so it would be louder. It would be even louder than the air conduction. BC is like basically bone conduction and AC is air conduction. Does it make sense to you? Why would it be higher in conduction deafness? Because you don't have any background noise, so now the bone conduction would be heard even louder than in a normal person. Okay. Now the other test is called Weber's test. Now again, they would place a tuning fork and the vertex in the midline. Now when it's placed, normally, this is like bone conduction, it's going to be heard in both sides, like normally. In both sides, it's going to have the same loudness, let's say, or equally. If the person has unilateral conduction deafness, the sound is going to be lateral on the it's going to be louder in the affected ear. Why is it louder in the affected ear? Again, same concept, because we don't have any background noises. So the bone conduction is going to be louder in the affected ear. Like let's say this person has a conduction deafness in their right ear here. This is the right and this is the left. If someone has conduction deafness in the right ear, then the sound would be even louder in the right ear. And this is because there's no background noise. They can perfectly hear the bone conduction. However, if there is nerve deafness, then one sound is going to be heard on the normal side. And if someone has a nerve deafness, let's say on the left side, then obviously it's going to be heard higher on the right ear, because nerve deafness meaning the person cannot hear aslan to begin with, whether it's bone conduction or air conduction. Clear so far? Do you understand them before we compare? Yes, okay, perfect. So normal Weber test where we place the tuning fork on the vertex of the skull, there is no localization. Localization meaning the sound is heard on one side more than the other. So normally there would not be any localization. If there is a conductive deafness, then it's going to be localized or heard louder on the affected side because there's decreased transmission of background noise, so we can hear the bone conduction even louder. If someone has sensorineural deafness, it's going to be localized to the unaffected ear, or we're going to hear it louder on the unaffected ear. Let's say, for example, this is the right, and this is the left, and if someone has, for example, sensorineural deafness on the right, then the sound is definitely going to be higher on the left because there's decreased transmission of all sounds in case of sensorineural or neural deafness. Now, Rennie's test that we talked where we place the tuning fork and we measure the air conduction and bone conduction. In a normal person, the air conduction is louder than the bone conduction. If someone has a conductive deafness, then again, the bone conduction would be louder for like the same reason that the transmission, decreased transmission background noise. If someone has sensorineural deafness, then the air conduction would be louder than the bone conduction. Okay? Now we're going to talk about the condition that happens in elderly, which is called a presbycusis. Now, presbycusis is a gradual deterioration of the spiral organ of corti, which is the sensory organ for hearing. I mentioned before, generally in elderly, almost everything in the body starts to degenerate, and one of which is going to be the organ of corti, primarily like the hair cells. So this will, because the hair cells are damaged, this will cause sensorineural deafness, because the hair cells are not transmitting the sound. They're the structures to hear to begin with. It's not for transmission, it's for hearing. So that's why we don't call it conductive. It's considered a sensorineural deafness. And it's very common in elderly, but now because of the loud world, loud music, headphones, and so on, it's going to be, it's becoming more common in even younger individuals, and it's progressive and cumulative. So it doesn't like happen overnight. It's not traumatic. It's like throughout the year, the hearing becomes lower and lower and lower until it reaches the deafness. Okay. Does anyone have a question before I move on to the questions? Clear. Okay, perfect. So can you please answer this question? Take your time. Okay, so 3 of you said A, and the answer is correct. It is A, because we mentioned in the sensory coding that the high frequencies are best heard at the base of the cochlea, or like at the base of the basilar membrane. And then at the apex, we hear the lower frequencies. So it is correct. Perfect. So it is A. Now, another question. Take your time to... Now, this needs thinking, and it's an information that I've mentioned. Okay, so I have B, C, A. Okay. So this is actually a question that came in our final. So it's from 14th batch. The correct answer is B. Well, the key word here, so the patient faces difficulty hearing in loud places. Okay, this can be anything. It can be literally due to damage of any of the structures in the ear. But he also hears sounds of females lower than males. Well, now let's think about it. I've mentioned like more than one time that the females sounds have a higher pitch or higher frequency. And we just said in the previous question, like literally it's asking you about the same thing, just in a different way. That the higher frequency, meaning the female sounds, are heard near the base of the cochlea. So because it's a female sound that are heard lower, it's gonna be okay. Like saying he thought no, he hears females better. Okay, now it kind of makes sense why I didn't choose B. But B is like asking you, the female sound are lower, meaning the higher frequencies are not heard properly. So which structure is affected if you can't hear the high frequency sounds or high pitch sounds? It's going to be the basilar turn of the cochlea. So the answer is B. Now A, C, D, they all can cause like decreased hearing, but no, there is nothing in the question that points towards them. Whereas hearing the sounds of females lower, that's the basilar turn of the cochlea. And this is from previous batches, so yeah, understand the concept, okay? Do you understand why it's B? Like all of you? Yes? Okay. Okay, so now we're gonna move to the next question. Have we A? Okay, so sorry. Someone asked me if we talked about A and D, or have we studied A and D? A, yes, we did. The helicotrema is basically where the perilymph of the scala vestibuli joins with the perilymph of the scala tympani. D, no, we didn't study it yet. Okay, so back to this question. Take your time to answer. Okay, all of you said D, and it's the correct answer. So a sincere neural cause of deafness is going to be presbycusis because perforated eardrum and middle ear ossicles and, and otosclerosis, they're all examples of a conductive deafness because you're not transmitting the sound. However, presbycusis, we said no, there's going to be degeneration of the organ of corti, so it is a sincere neural cause of deafness. Okay. So now we're going to move to the anatomy of the vestibule and the semicircular canal. So now we're going to dive into the balance and the equilibrium parts of the ear. And it's the last part. So we're going to talk about the anatomy, which is very brief, and then the physiology, and we'll be done. Inshallah. Okay. So vestibular apparatus maintains the body, mainly the head. So the body overall, like your hands, your arms, your legs, so on, like the equilibrium is mainly maintained on the cerebellum level. It's not on the vestibular apparatus. The vestibular apparatus is mainly about the movements of the head itself. So other parts of the body, the vestibular apparatus is not really that involved. It is to a certain extent, but it's mainly important to the head, okay? At equilibrium and stabilizing the eyes relative to the environment. We'll talk about this in more details. Now, when it comes to equilibrium, we have two types of equilibrium. We have the static equilibrium and the dynamic equilibrium. Now, the static equilibrium is like maintaining the position of the body relative to the force of gravity. Basically, what it's trying to tell you is that the body itself is not like rotating. It's just you're maintaining it relative to the force of gravity. Like, for example, you're in a car and then you're speeding the car and then suddenly the driver hits like the brake and the car stops. So there'd be some sort of like resistance to any change of movement, and by that you would go like forward and backward. That's basically your body trying to maintain your static equilibrium, so you're not rotating. Dynamic equilibrium, so it's mainly related to rotations, accelerations, and decelerations. So these type of movements is what we call the dynamic equilibrium. Now, the static equilibrium is the function of two organs that we're going to talk about that are called utricle and the saccule. And the dynamic equilibrium is a function of an organ, or three actually, semicircular canals. We're going to talk about them more later. Just know that the utricle and the saccule collectively are known as the otolithic organs. We're going to talk about it more. And then we have the semicircular canals. Okay. Now, the vestibular apparatus is basically the structure here. So we have the cochlea. I'm sorry. I'm so sorry. The vestibular apparatus is this structure here. I missed through. Okay, so this is our vestibular apparatus. So we have the utricle. This structure here is the, sorry. We have the vestibule where we have the saccule and the utricle, and we have the semicircular canals. So we have a superior semicircular canal. We have a lateral semicircular canal. This should be posterior, I believe. This should be posterior, shouldn't be lateral. I don't know why it's lateral, but it should be posterior. So this is the superior, this is the posterior, and this is the horizontal, which is also known as the lateral semicircular canal. Now, the vestibule has the utricle and the saccule, which we said we refer to them also as the otolithic organs. Now, generally speaking, we have something that is called the bony labyrinth and the membranous labyrinth. This is also present in the cochlea. Remember when we talked about the bone, the modulus, that's considered basically the bony labyrinth, and then we had the membranous labyrinth, which is mainly the scala media where we have the endolymph, okay? So a very similar concept is present in the vestibular apparatus. So we have the bony labyrinth, which surrounds the membranous labyrinth. Now, the membranous labyrinth, always, like keep it in your mind, the membranous labyrinth is the one that is filled with endolymph. And we said that the endolymph is the fluid that is rich in potassium. And between the bony and the membranous labyrinth is where we have the perilymph, which is high in sodium, low in potassium. But what's the bony labyrinth when it comes to the vestibular apparatus? The bony labyrinth is the vestibule, which is this structure here, which contains the utricle and the saccule, and the semicircular canals. Focus on the word canals here. So these vestibule and semicircular canal are kind of the bony parts of it. And then inside them, we would have the membranous labyrinth. Now, the membranous labyrinth is going to be the utricle, the saccule, and the semicircular ducts, not canals. So the ducts are present inside the canal. The canal is considered the bony labyrinth. Inside the canal, we'd have the ducts that are considered the membranous labyrinth. So inside the ducts, we would have the endolymph. In between them, we would have the perilymph. Now, looking at the structure, so we were in the cochlea, we said it's mainly for hearing. And then the cochlea would be connected to the saccule. And then the saccule would be connected to the utricle. These collectively are known as the vestibule, and we said they're responsible for balance and equilibrium. The utricle and the saccule are membranous labyrinth, whereas the vestibule itself is the bony labyrinth. Then the utricle is connected to the semicircular canals. We have the superior, the posterior, and the horizontal canal. Inside the semicircular canals, we have the semicircular ducts that are considered parts of the membranous labyrinth. Does that make sense? Do you understand the anatomy so far? Yes, okay, perfect. So now we're going to talk about the utricle and the saccule, which is these structures here. We were kind of going to zoom into them. So the saccule is connected to the utricle viaduct. As we said, they're connected. Now remember the organ of Corti that we talked about in the cochlea, and it's the sensory organ for hearing in the cochlea. Now the sensory organ, in case of the vestibule or the saccule and the utricle, actually, to be exact, the sensory organ is something that we call the macule. Now this macule is present on the medial wall of the saccule. Now look at this picture, it's going to make it easier. So it's on the medial wall of the saccule, and it's present in the floor of the utricle. So this is the utricle. You see in the floor, we're going to have the macule, and the saccule, we're going to have the macule and the medial wall. Now, what is the macule exactly? The macule is very similar to the organ of Corti. So it is basically the hair cells that are embedded in a gel-like structure. But what's special here in the macule, in the saccule, and the utricle is that we have these otoconia. Now, what is an otoconia? Otoconia are calcium carbonate crystals. So these otoconia are calcium carbonate crystals with the gel, with the hair cells. This collectively is what we call the macule, and it's present in the saccule and the utricle. Okay, does that make sense? Like, do you understand the idea of utricle, saccule, and then the sensory organ of the macule? Okay, perfect. And it's important for static equilibrium, because we said we had static, I'm sorry, 1 second, but I'll just, okay. Sorry for the notifications noises. Okay. So we have the utricle and the saccule. Now the utricle, we said, the macule is going to be present in the floor, and the saccule is going to be in the medial wall. Now these are important for static equilibrium. So we have two types of static equilibrium, either linear or vertical. Remember the example of the car that I talked to you about, where the person would move forward and backward to kind of resist the change in the equilibrium or the state that they're at. Now this forward and backward movement is what we call linear static equilibrium. So under the static equilibrium, we have linear and vertical. So the linear static equilibrium is the function of the utricle, which makes total sense to us. If you look at this picture and you're looking at the structure of the utricle, it makes sense that the forward and backwards movements are basically organized at the level of the utricle, because the utricle is kind of horizontally oriented. So when you're moving backward and forward, the utricle would be the one that is responsible for the equilibrium. Now, the vertical equilibrium is the function of the saccule. An example of the vertical equilibrium would be the elevator. Now, when you're in the elevator, you're changing your position, and your body can sense that you're going upwards and downwards. Now, what maintains your equilibrium at this state would be the saccule, and it makes sense, again, if you look at the structure of the saccule. So there's like up and down movement, or vertical and downwards movement. So that's vertical static equilibrium is organized at the level of the saccule. Does that make sense to you? And do you understand why it's one is in the utricle and one is in the saccule. Yes, okay, perfect. Now we're going to take the utricle, which is this one, and we're going to kind of go in more details. Now, exactly the same concept as the organ of quartzi. So we have the stereocilia, which are present on top of the hair cells, and then we have one kinocilium, which the stereocilia would bend either towards or against. And we have this gel-like structure. But what's different here is the presence of these autoconia. Autoconia are these calcium carbonate crystals that are present on top of the autolytic membrane, or this like gelatinous-like structures on top of it. Okay. That's it for the utricle, and we said that it's responsible, like sensitive to horizontal movement, like the movements in the core again, or basically what we call as the linear equilibrium. Now, when it comes to the saccule, it's flattened, irregular shaped membrane, again, very similar to what we said. It has the same structure as the utricle, however, it lies perpendicular and vertical to the macule of the utricle. So yeah, as you can see here, it is perpendicular and vertical. So the saccule as compared to the utricle makes total sense. And there isn't much to explain here. I'm just trying to find if there's anything that's worth explaining. Okay, so now when it comes to the saccule, we have two openings. We have one opening that's called the endolymphatic duct, which communicates with the utricle, because we said earlier that the saccule communicates with the utricle, and this communication is done through this endolymphatic duct. So the name of the duct that is connecting the saccule to the utricle is the endolymphatic duct. Now, the other opening that communicates with the duct of the cochlea is the ductus reunions. Ductus reunions, I think. I honestly did not practice how to pronounce that, and I don't remember it in second year at all. But that's a duct that is communicating with the cochlea. Because we said cochlea, it's like communicating with the saccule, and then the saccule communicates with the utricle. If you look at the picture here. So we said cochlea communicates with the saccule. So this is the ductus reunions. And then the saccule communicates with the utricle through the endolymphatic duct. Okay? So that's it mainly. Does that make sense so far? Like, do you understand everything I've said earlier? Okay, perfect. Now we're going to talk about, honestly, I'm just going to touch upon them briefly. I don't think they're going to ask you at all, just for you to understand the concept. There are a lot of pathways that are involved in maintaining our equilibrium and balance in general. So we talked about it in neuro, like the proprioception. So you know where the position of your limbs and muscles are. So that's one pathway to maintain your equilibrium. So knowing where you are exactly in terms of joints and muscles. So that'll be your proprioception. Or your eyes, whatever you can see in front of you can also tell you about the position of your body or where you are, or impulses also from the cerebellum. So the cerebellum receives and receives the inputs and also modulates your movements accordingly. All these movements are going to be taken to the vestibular nuclei. And apologies, remember I said the vestibular nuclei is about like the balance and cochlear for hearing. So that's the vestibular nuclei. They're going to be transmitted to the oculomotor center of movement to the eye and the spinal cord for correcting the balance of the body. So it's just giving you the overall concept how everything is connected in our body to maintain our equilibrium and balance. So everything you studied in neuro and everything in head and neck. So just trying to give you the full picture. So we're done with the vestibule and the utricle and the saccule. Now we're going to move to the semicircular canal. Now we said their importance in dynamic equilibrium or rotational acceleration. So when the person is rotating, what's maintaining our equilibrium at this point is going to be the semicircular canals. Now they're projected in 3 different planes. So there's the X, Y, and Z axis. All these planes are covered by the semicircular canal, subhanAllah. It's actually really, really interesting when you study the physiology of the balance. Now, let's look at the semicircular canal here. So we said we have the superior, the posterior, and the horizontal or the lateral semicircular canal. Inside the semicircular canal, we have the semicircular ducts, which are part of the membranous labyrinth. And at the end of the semicircular ducts, we have this kind of dilation, which we call the ampulla. This here. Now, inside the ampulla, we have the sensory organ of the semicircular canals, which is called the crista, or sometimes also known as the crista ampullaris, because it is present inside the ampulla of the semicircular canal. Now, you really have to know which sensory organ is present in which part of the inner ear. So the organ of Corti is in the cochlea. We said the macule is present in the utricle and the saccule, which are present in the vestibule. Now, the last sensory organ is the crista ampullaris that is present in the ampulla of the semicircular canal. And it's important for rotational acceleration or dynamic equilibrium. So far clear? Like, do you understand everything I've said so far? Yes, okay, perfect. Now we're going to dig into the physiology. So the anatomy of them is very easy. Like, there isn't much to talk about in the anatomy. So we're going to go to the physiology to understand it better. I'll also touch upon the anatomy again in the physiology part, so that's, we have a full lecture. So we have three main sensory systems that are responsible for our spatial orientation and posture. So the first one is going to be that we talk, the main or lecture is going to be the vestibular system, which is made of, the vestibule, utricle, and saccule, and the semicircular canals. The other two are the visual system, so whatever you see, like coming from your retina, and the somatosensory system that conveys the peripheral information from skin, joints, and muscle receptor, which under the somatosensory system would have our proprioception and like the awareness of our limbs and joints or where they're present. These systems are stabilizing our body and they overlap to compensate for each other's deficiency. If someone, la samah Allah, lost their vision, they wouldn't entirely lose their equilibrium because of the vestibular system and because of their somatosensory system. They still can, they still have proprioception knowing where they are. And vice versa. If someone has any defect in one of them, two would compensate. And you'd understand this concept more in pro, where there are tests that tell you, if someone has only one that is defected, it's fine. It wouldn't really show unless you perform a certain test to cover, then it would show even more. I'm not gonna overwhelm you with that, but just understand the concept that the three are covering for each other. Now, the gravitational pull and rotational movement are stimuli for vestibular apparatus, which makes total sense again, because we're trying to stay in an equilibrium status. So if there's any rotational movement which deviates us from the equilibrium, we'll activate the vestibular apparatus in order for us to remain in our equilibrium. Okay, so it provides information about the movements. Okay, basically just everything we've said before. Now we said the sensory receptors in the semicircular canal is the crista, and the utricle and the saccule, the sensory organ is the macule, and in the cochlea it's going to be the organ of Corti. Now, if there is any damage in one of these sensory organs, crista or macula, damage to these damage to these sensory organs can result in head tilt, vertigo, and nausea because they're mainly responsible for our balance, our equilibrium. If there's any damage to them, then there would be loss of damage. There would be loss of our equilibrium. There would be vertigo. Vertigo is basically when you feel that the world around you is rotating. So because you've lost your equilibrium, so that's what the person would experience. Okay, they're present in the petrous portion of the temporal bone. Okay, which is basically everything in the ear and almost everything the tympanic brain's attached there. So majority of the inner ear is present in the petrous portion of the temporal bone. And then there's the membranous, which is the membranous labyrinth that we discussed, the cochlear ducts, not the canals, and the utricle and the saccule. Okay, that's everything we discussed already. Now, what is equilibrium? I keep saying equilibrium. So it is a state of balance. So you position your body in a kind of a stable state, despite that you have gravitational conditions or any movements, even in the car. So you stay still because of your equilibrium. Even though the world around you is changing, whether the car is moving, the plane is going upwards, your body stays in the same position due to the equilibrium status that the body's constantly trying to reach. Now we said that there's dynamic components and there's the static components. Now the static, we said there's either linear or vertical, and the linear was the function of the utricle and the vertical was the function of the saccule. And they're all detected by the hair cells, and it's the exact same concept of bending of the stereocilia towards the kinocilia, opening of the potassium channels, and so on. Okay, so that's everything we've discussed. Again, we've discussed that. That's again the anatomy that we've discussed. Okay, so now I'll just talk about the planes in which the semicircular canals are present, or the movements. Now, each movement of our head, rotational movements, a certain semicircular canal is responsible for it. So if I'm tilting my head and saying no, that would be the function of the posterior canal. So the posterior canal is mainly responsible for the head tilt. I'm sorry, the head tilt is this. Sorry. The head tilt is doing this when the head is tilting. That would be the posterior canal. And then the superior canal is nodding or saying yes. So that would be the superior canal, because it makes sense. If you look like how it's oriented, it makes sense for the tilt to be in the posterior and the nodding to be in the superior canal. Now, when it comes to the horizontal canal, which is placed like horizontally, now this one is the one that is responsible for like shaking your head or saying no. The tilt is the posterior canal. So that's the different function of each semicircular canal. Now on the x-axis, we're gonna have the horizontal canal, which makes sense. This is our x-axis. The superior or the anterior semicircular canal, we're gonna have the y-axis, and on the z-axis, we're gonna have our posterior canal, which again makes sense if you look at how they're oriented. By the way, you should know which one is anterior and posterior and horizontal. This might come in OSPE, so just know them. Like this is something you just ignore, but you have to know which one is anterior, posterior, and horizontal. So now the anterior semicircular canal, it makes a 45-degree angle with the posterior semicircular canal. Now the posterior semicircular canal again forms a 45-degree angle, so it's kind of, they're like perpendicular to one another, but not literally. Now the horizontal semicircular canal, it makes a 30-degree with the horizontal plane, which kind of makes sense. You don't need to know it in that details. Honestly, they're never going to ask you. Now, a very important concept when it comes to the semicircular canal is that the posterior, whenever we activate, for example, the posterior semicircular canal in one ear, we're gonna unactivate it in the other ear or deactivate it in the other ear. This is a concept that I want you to understand, because once we talk about like different tests, like later in the lecture, I want you to understand this concept, that if we activate one semicircular canal in one ear, we immediately deactivate the one in the other ear. Like this is how our body works, so that you don't get conflicting signals. Like for example, if one semicircular canal tells you that you're rotating to the left, the other khus would not tell you that you're rotating to the right, for example. So it's going to be just deactivated. So the posterior canal shares plane with the contralateral anterior canal, and the horizontal canals share the same plane. Okay. Now talking about the semicircular canals and not the ducts, meaning inside the membranous labyrinth. So remember how the semicircular canals we talked, they end up having this dilation that has the ampulla, and then we have the sensory organ, the crista ampullaris. Well, this is it. This is like a zoomed in picture. Now, the crista ampullaris is again a very similar structure of like the organ corti and the saccule and the utricle. The difference here is that we have the hair cells, and on top of the hair cells, we have this gelatinous structure that is called the cubula. So whenever we each one of them has a membrane, basically. So the membrane here and the crista ampullaris, or the crista, is what we call the cupula. And it's present in the ampulla of the semicircular canal. Yes, now, okay, a very important concept to understand here is the inertia. Now, inertia it's the characteristics of objects whenever the object is trying to kind of resist any change of movement. So let's take the water as an example. Let's imagine, I want you to imagine it in your head that you have a bottle of water that you're bending forward or like pushing forward. When you're pushing it forward, initially, the water would go backwards. And that's because the water, like it's just a characteristic of subject to resist the movement forward. So the water would just go backward while the bottle of water is going forward. Like, can you visualize that? Can you see it in your head? You know how the water would just go backward when you're pushing the object forward? That's due to inertia. Now, this characteristic is also present in the perilymph and the endolymph that are present in our ear. So whenever you start like any kind of rotation or activation of the semicircular canal and you're moving it towards any direction, initially the endolymph would move in the opposite direction. Why does it move in the opposite direction? Because of inertia. So for example, you're rotating your head to the left. Okay, let's say you're rotating your head to the left. The endolymph would move to the right, and that is due to inertia. So it's trying to resist any change in the movement. Now, when the endolymph moved to the right, in this case, for example, it will cause bending of the stereocilia in the direction of the kynocelia, and this would open the potassium channels in the tip links, causing depolarization, opening the voltage-gated calcium channels, and then release of the neurotransmitter. So very similar mechanism as like the hearing and everything, but the difference here, is the stimulus mainly, it's due to rotation, and that the concept of the inertia that is present in the endolymph here. Like, does it make sense to you so far? I can go over the physiology in more details, but I've already explained it in the organ of corti, and like there's nothing different, basically. Okay. Okay, perfect. Again, this was just trying, I forgot to like put the one that's animating, but you can play it in Dr. Yaqeen's lecture. Basically, it shows you how when you rotate your head to a direction, basically the endolymph would move in the opposite direction initially, but due to inertia. Okay, so the otolith organs are the utricle and the saccule, and we call them otolith organs because we have the otoconia, which are the calcium carbonate crystal. We said the utricles are horizontal, the saccule are verticals. They have a otolithic membrane covered with calcium carbonate. That's everything we discussed also earlier. Bending of the stereocilia opens the potassium channels, releasing the neurotransmitter. They're not sure what neurotransmitter is being released, but no, some research articles said glutamate, and like it's not really finalized yet, but we know hearing it's definitely glutamate. And it's the nerve here that is connected to the hair cells, it's going to be the vestibular nerve. Now, when it was hearing, it was the cochlear nerve. When it comes to balance and the ones that attach to the hair cells in the otolith organs, it's going to be the vestibular nerve. Okay, that's again what I said. I'm sorry, I forgot to edit the slide. Okay, so basically it's just trying to show you what the central connections of the vestibular system is. So we have lots of connections. So it's connected to the thalamus, to the spinal cord through the vestibulospinal tract, and like everything we've talked about earlier. And it's connected to the vestibular nuclei. So we have superior, medial, lateral, and inferior vestibular nuclei. It's also connected to the brainstem. It's going to go to the medial longitudinal fasciculus and reticular formation. Now, a very important thing in the reticular formation is that it's associated with the nausea and vomiting. It also coordinates the eye movements. We're going to talk about this in more details later. Is here the reticular formation afferent to the vomiting center and motion sickness. Okay, so basically everything we've discussed, I don't think they're going to ask about anything from this slide. I just wanted to give you an overview. I'll edit the slide before sending it to you, inshallah. I just forgot to edit it. Anyway, now we're going to talk about the vestibulo-ocular reflex. Now, I want you to all, now that you're watching me, I want you to all focus your eyes on your laptops or iPads or whatever device you're using. So keep your eyes on your laptops, okay? And stay in the center. Now, I want you to all rotate your head to the right side while keeping your eyes on the laptop. Okay, so focus on the laptop and rotate your head to the right. Now, because you're rotating your head to the right, automatically, your eyes would go to the left. See, like I'm focusing on my laptop right now. When I rotate my head to the right side, my eyes would automatically go to the left side. Do you see that? Like, do you see it initially when you're focusing on an object? I want you to know if you understand this concept. Like when you're rotating your head to one side, how the eyes would move to the opposite side. So my head is rotating to the right, my eyes would go to the left. Okay. So this thing that I just did here, if you have an intact vestibulo-ocular reflex, then you should have experienced the same thing. When your head turns to the right, your eyes rotate to the left, not necessarily right and left. It could be like the opposite direction. Basically, the concept, and when you rotate your head to one side, the eyes would rotate to the opposite side. This is like the purpose of vestibulo-ocular reflex is to maintain fixation on the original image or the target that we were staring at. But after a while, which now this nystagmus is what we call the slow phase. Nystagmus is basically kind of involuntary eye movements. So I involuntarily, my eyes moved to the left while I'm just staring at my laptop. So I didn't move my eyes to the left. No, I was just staring at my laptop. Automatically, my eyes just moved to the left so that I keep staring at my laptop. So that's what we call the slow phase of nystagmus. Now, let's imagine if my sister came from my right and called me. So initially, I would have this slow phase, then I would correct my eyes towards my sister to look at her. Well, that would be the fast phase of nystagmus, where I would correct my eyes to the right so that I move from staring at the laptop to looking at my sister. Now, the concept in nystagmus is your body doesn't want, sorry, the vestibulo-ocular reflex in general, not only like nystagmus, the body doesn't look throughout the whole environment while you're changing, for example, your head movement. The body doesn't like it this way. So when you're changing your eye from one object to another, you would have a vestibulo-ocular reflex that you don't feel aslan that you have it. So your eyes would move from one object then to the other. So you don't rotate your head like with every single head rotation. You don't, move your eyes with every head rotation. Does it make sense? Like, do you understand why do we have the vestibulo-ocular reflex? So you don't move your eyes like with every time you rotate your head. Yes, okay. Does anyone want me to repeat anything? Is there anything that anyone does not understand so far? All good? Okay, perfect. So we said we have the slow component, which you're tracking an object or you're staring at it. And the fast component is going to be in the opposite direction, where basically I'm correcting my movements back to my sister's example. Like if my sister came from my right, I would initially have the slow phase where I'm still staring at my laptop and my eye is on the left side. Then I would correct my eye in the fast nystagmus, looking at my sister. Fast component is in the direction of the head movements, because my head initially tilted to the right, then I corrected my eye to the right side. Now, the slow phase is due to excitatory and inhibitory input from the neurons to the motor neurons controlling the eye movement. And the fast components are excitatory and from the pontine reticular formation. So this one is like from neurons in the semicircular canal. This one is in the higher level, from neurons in the reticular formation that are controlling the eye movements. Okay, this is again everything I explained, but in different direction, whether it's right or left. And then this is like the opposite direction, and this is upwards and downwards. Now we're gonna move to a test that is done in the clinic, which is called the caloric test. Before I start with the caloric test, basically, I want you to understand one thing, that the warm water or the hot water stimulates the ear. The cold water inhibits, or it does not stimulate the semicircular canal. So the hot water is excitatory, the cold water is inhibitory. Now we're going to talk about the caloric test. So what the clinician does, or the doctor, basically they would apply hot water on the ear if like, for example, if the patient's unconscious and we want to check the function of their like medial lemniscus, we're going to talk about medial lemniscus later, we're going to check the function of their brainstem or whatever. So we would apply hot water in the right semicircular canal. Now we said the hot water is is excitatory or stimulatory. So the body would perceive the hot water in the right ear as if the head is tilting to the right. This is how the body perceives it, because the patient's unconscious, they don't know. When you place hot water, it stimulates the right semicircular canals. This will be perceived in the body as if the head is rotating to the right. So because of the vestibulo-ocular reflex, the eyes would move to the left. Because we just placed hot water, so we said vestibulo-ocular reflex, so the body would perceive it as if the head is moving to the right. So you're going to see the eyes of the patient, like the patient would usually be unconscious. You're going to see the eyes of the patient moving to the left side. This indicates that the vestibulo-ocular reflex in this patient is intact. There's no brainstem damage usually. And they're more accurate tests for brainstem, to be honest, but to understand the concept. Or there is no medial lemniscus damage. We're going to explain the medial lemniscus later. So it demonstrates the vestibulo-ocular reflex, who helps in maintaining the visual stability during the head movements. Now, the cold water is the opposite. Remember the concept that I told you, if we stimulate one ear, we inhibit the other, and if we inhibit one ear, we stimulate the other? Now, this is where this concept applies. So if you apply the cold water on the right ear, you're inhibiting the right ear. And by default, when you inhibit the right ear, you stimulate the left ear. So now you're going to stimulate the left semicircular canals. So the body is going to perceive it as if the head is being is rotating to the left side. So when the head is rotating to the left side, the eyes would move to the right. That's the the slow component. So the slow movement, and the eye is going to move to the right because we applied cold water. We said it's inhibitory, so we're going to stimulate the semicircular canals on the other ear. So the body would perceive it as if the head is rotating to the left. That's if we apply the cold water in the right ear. So as if we rotated our head to the left, so the slow phase, that the eye would move to the right, and then the fast phase, where the correction, the eyes would move to the left. Usually the correction is not present in unconscious patients, but in like a healthy patient, you would also see this eye moving to the left and then to the right and so on. This also illustrates the vestibulo-ocular reflex, where information from the inner ear to adjust the eye movement. Do you understand the vestibulo-ocular reflex and the caloric test before I start comparing? Okay. Okay, so I'm not sure if sorry. Okay, I don't want you to see the answers, but I forgot to add the slides of the medial longitudinal fasciculus. Okay, so we have a structure in our brain that is called the medial longitudinal fasciculus, which is very important when it comes to our eye movement. Now, let's go back to my example where I said, I've rotated my head to the right and I moved my eyes to the left automatically because I'm staring at my laptop. Now, can someone tell me, now that I'm moving my eyes to the left, I want you to take your information from neuro and apply it here. When I moved my eyes to the left, which muscle was activated on my left eye and which muscle was activated on my right eye? The eye muscles. What caused this movement of my eyes? Did you take this in neuro? I think you did, صح? Like one is innervated by the abducens, one oculomotor nerve. Yes, exactly. So we have the lateral rectus muscle and the medial rectus. So on my left side, it was the lateral rectus that is moving my eye laterally. Whereas on my right eye, it was the medial rectus that is moving my eye medially. So this information is very important when it comes to the caloric reflex. Because if the person has an intact caloric reflex, meaning this means that their abducent nerve is intact because they can move their eye laterally. Let's say, for example, I'll keep the right and the left example. My head to the right, my eyes to the left. So this means my abducent nerve is intact on this side. My oculomotor nerve is intact on this side because this is the lateral rectus, which is innervated by the abducent nerve. And this is by the medial rectus, which is innervated by the oculomotor nerve. Now, I don't know if you remember the medial longitudinal fasciculus, which was coordinating this movement. I'll actually add a slide because it's very hard to explain it without a picture. But the medial longitudinal fasciculus is what's connecting the abducent, the oculomotor, and the trochlear nuclei together to coordinate the eye movements. Like, do you remember the structure? I'm sure you've taken it in neuro, just not as details as you'll take it in head and neck. But either ways, I'll add a slide to make it easier. Okay, just 1 second. I'll stop sharing. I'll add a slide and then we continue. Okay. Excuse the quality of the picture. Inshallah, I'll find a better picture when I send you the PPT. But I want you to understand the concept is that at the level of the brainstem, we have all the nuclei of your oculomotor, trochlear, and abducent. The trochlear is not really as important, but it's also contributing in the medial longitudinal fasciculus. So basically, the information that are going from the oculomotor nucleus to your medial rectus and from your abducent nucleus to the lateral rectus, they're all coordinated at the level of the medial longitudinal fasciculus. So if the medial longitudinal fasciculus is impaired, you're going to see that the patient would present to you in a lesion that is different than when it's a brainstem lesion. So let's see how they're impaired, and I'll explain in each lesion. So I'll use the lesions to explain. So a normal person would have my reaction. So I moved my head to the right, so my eyes moved to the left. That's also seen in a patient where the brainstem is intact. Now the problem is if the patient has medial longitudinal fasciculus lesion bilaterally, so basically the lateral rectus is not going to be affected. It's only going to be the medial rectus. Why is that? Because the abducent nerve directly innervates the lateral rectus. However, the oculomotor nerve, it uses the medial longitudinal fasciculus to innervate the medial rectus. So if there is any lesion in the medial longitudinal fasciculus, then the medial rectus would not work because it needs the medial longitudinal fasciculus in order for it to like move the eye to the medial side. And the patient would only have the left movement on the left eye. Say, لا سمح الله, I have an MLF lesion, you're only going to see my left eye going to the left, and my right eye would stay in the center. So that's what happens if the medial longitudinal fasciculus is impaired bilaterally. I'll have this written in words when I send you the PPT, inshallah, with a better picture, because I forgot to add it. Now, if there is a lower brainstem lesion, the patient would not have a reaction to begin with. Nor in the left, nor in the right ear. So you'd move the head and the eyes would move like with you. They would not like have a kind of compensatory action where they move to the opposite side. So it's very similar to the warm water. You just apply what we talked about. So the warm water would stimulate the right semicircular canal. So the body would perceive as if the head is rotating to the right. So the eyes would move to the left. So that's also the case of the brainstem is intact. If the patient has a, if the patient has a medial longitudinal fasciculus lesion, then you're going to see that the medial rectus is the one that's going to be affected, not the lateral rectus. Why? Because we said the abducent nerve directly innervates the lateral rectus. However, the oculomotor nerve innervates the medial rectus through the medial longitudinal fasciculus. And in a brainstem lesion, the patient would not have a reaction to begin with. Did you understand the concept? Don't worry about the tract. I'll add a better picture. I couldn't find, honestly. I'll just look more for a better picture, and I'll have it written in words, inshallah, when I send you the PPT. But do you understand it so far? Like, do you understand the concept of the caloric reflex and the vestibular ocular reflex? All clear? Perfect. Okay, so now we're going to move to the benign paroxysmal positional vertigo. Now we know that in the utricle and the saccule, we have these autoconia, which are the calcium carbonate crystals, and they should normally only be present there. Now, if someone has a dislodged otolith or the calcium carbonate crystal from the utricle, and it bumps into the capula, because if you look at the anatomy, it would make so much sense. Let me just, okay. So basically, the otolith is going to be dislodged from the utricle, and it's going to go to the capula and the ampulla and stimulate the hair cells in response to the head movements. Let's go back to the anatomy so that you kind of can, oh, actually I have a picture here. So we know that the utricles are connected to the semicircular canal. So we have like the saccule, then the utricle, and then the utricle are connected to the semicircular canal of the otolith or the otoconia are dislodged from the utricle, and they go to the semicircular canal itself, or to the ampulla or the crista ampullaris, which is part of the semicircular canal. If they're dislodged and they go there, this is what can cause benign paroxysmal positional vertigo, because the body would keep perceiving signals as if the head is turning, even though the head is not turning. And like the visual information is telling you that no, you're not turning, but the body is sensing that you're turning. So the body would receive conflicting information, because the otolith or the calcium carbonate crystals are stimulating your semicircular canal, but your sensory, your visual information and your somatosensory information, they indicate otherwise. So the conflicting information causes vertigo to the person. And this is one of the most common causes of vertigo. You're going to talk about it more in your clinical lectures, but this is just a brief introduction. And you should know the pathophysiology is the otolith being dislodged from the utricle. And it commonly goes to the posterior semicircular canal, which makes sense again, because it's the one that's kind of on the same plane as the utricle. Does it make sense so far? Like, do you understand everything I've explained? Yes, okay, perfect. So I want you to answer this question now. Okay, so I have 4 B's and 1 A. The correct answer is B. That's because the saccule, if you remember, we said it's kind of vertically placed, so it is the one that is responsible for our vertical movement, like in the elevator. However, the macula and the utricle is mainly responsible for like horizontal equilibrium, like moving forward and backwards, not the vertical parts. Okay, now that's another question. Take your time answering it. Okay, so I have 4 Ds, and yes, D is the correct answer. Since you all answered it correctly, I don't think I need to go through the other MCQs. If someone is watching the recording and they don't get why it's not any of the options, you can email me. So one is asking why is not E. Inward movement of potassium through the voltage-gated potassium channels in the stereocelium membrane has depolarizing influence. Because the potassium channels are not voltage-gated. The potassium channels that are present are basically mechanically gated. So when the stereocilia bends towards the kinocilium, the potassium channels would open. They're not voltage gated. The ones that are voltage gated would be the calcium channels. So it's not a voltage gated channel. That's why it's not E. Does it make sense to you? Like, it's not a voltage gated potassium channel. It's a mechanically gated potassium channel. Okay, perfect. If you have any question, whether in my slides or in the slides of the doctor, of course, please feel free to email me. My email is in the first slide. And please scan the QR code. I'm looking forward for your feedback. If you have any questions, please email me, or even if you have my number, you can freely contact me on WhatsApp. And thank you. I hope, inshallah, you benefited. And that's it. Good luck studying head and neck, and good luck in your finals.